അവിടെ നിന്നുള്ളവർക്ക് കിട്ടില്ല. ഈ ലൈറ്റ് ഓഫ് ആക്കാം. വീഡിയോ അവിടെ പ്രോബ്ലമാ? ശരി, ആയിക്കോട്ടെ. വോളിയം കൂട്ടണം. തീരെ കേൾക്കുന്നില്ല. Good morning, everybody. Today we are going to discuss two new topics: embryology and genetics and thorax. First, we shall discuss some relevant questions from embryology. Let us start from the first chapter in embryology, spermatogenesis. In which of the following stages of spermatogenesis the chromosome number is halved? Show your digits. Diploid number becomes haploid number. 46 becomes 23. When does this occur in spermatogenesis? Number 2. Okay, we shall see. Look at this picture. This is the primordial germ cell of male reproductive system, spermatogonium. It undergoes several mitotic divisions, multiplications to form the primary germ cell, primary spermatocyte. Undergoes several maturation divisions to form several primary spermatocyte. Now the first meiosis occurs. The primary spermatocyte divides into two secondary spermatocytes with a reduction in the number of chromosomes. Now diploid has become haploid to form two secondary spermatocytes. Each secondary spermatocyte again divides. Second meiosis occurs. That results in the formation of 4 spermatids, each secondary without further reduction in the number of chromosomes. So it is in fact a mitosis, not actually a meiosis. Meiosis means there should be a reduction in the number of chromosomes. But this second meiosis is in fact a mitosis without any reduction in the number of chromosomes. In this way, 4 spermatids are formed from a single spermatocyte. Now what happens? The spermatid undergoes structural transformation to form 4 spermatozoa. Each one is transformed into a spermatozoa. The entire process is known as spermatogenesis, and the last stage, that is structural transformation of spermatid into spermatozoa, is known as spermiogenesis. The whole process is spermatogenesis, but the last stage is known as spermiogenesis. What exactly happens during spermiogenesis? We shall see this picture. This is a spermatid, an oval-shaped cell with all cell organelles, usual cell organelles, all are there in it, like the Golgi apparatus, nucleus, mitochondrion, centriole, all such things are there, just like we see in any other cell. And this spermatid undergoes structural transformation. Elongation occurs like this, gradually elongates, and finally a spermatozoon with all parts develops. Our exercise is to know which part of the spermatozoon is contributed by which of these cell organelles. Question can be set. From the picture, it is clear that the Golgi apparatus becomes the acrosomic cap of the spermatozoa. It contains an enzyme. What enzyme? Hyaluronidase. That is essential for penetrating the zona pellucida of the ovum. The nucleus becomes the head of the spermatozoa, which contains all genetic materials, the 23 chromosomes, 23 molecules of DNA, 22 autosomes and 1 sex chromosome, could be X or Y. The mitochondria become the, before that this one has come, the centriole forms the proximal and distal rings of the tail of the spermatozoa. The mitochondria surround the middle piece to form the mitochondrial sheath. You can see the mitochondrial sheath here. So mitochondria provide the mitochondrial sheath of the tail of the spermatozoa, and this provides the energy for the motility of the sperm. So energy in the form of ATP is provided by the mitochondrial sheath, which is formed by the aggregation of mitochondria. So questions can be said, which of the following in a spermatid becomes the acrosomic cap of spermatozoa? 1, 2, 3, 4 given. Now you know it is the Golgi apparatus. Which of these forms the head of the spermatozoa? 1, 2, 3, 4. You know it is the nucleus. So that way we can set questions. Okay, now the mitochondria also comes. Shall go back to the question now. So actual division, the first meiosis, occurs when the primary spermatocyte divides into 2 secondary spermatocytes. A person, some seats are vacant here, some of you can come and sit here, 2 seats are there in front. Instead of taking too much strain, you can come this side. Come on. Help her to come out. Come. seats are there in front. Okay. A person is likely to be sterile when the number of healthy motile spermatozoa per ml of semen goes below. When will you call a person oligospermic, not able to fertilize an ovum? Number three. When the number goes below 10 million, the person remains sterile. What is the approximate time required for spermatogenesis to be completed and for the passage of viable sperm to reach the epididymis? How much time is required for the completion of spermatogenesis? 2 hours? 2 days. The person is left with no viable sperms in the testis due to change in the temperature, body temperature. Normally spermatogenesis occurs 2 to 3 degrees Celsius below body temperature. Here, because of increased temperature, spermatogenesis does not occur. That is why the person is left with no viable sperms in the testis. Approximately how much time is required for the spermatogenesis to be completed following this fever? Number 3, 2 weeks. Number 4, exactly more than 2 months, 64 days. Sometimes it can go up to 72 days. More than 2 months required for the spermatogenesis to be completed. This much time is required. More than 2 months. In physiology guideline given 72 days. Anyway, one thing is clear, more than 2 months. During which period of life the ovary contains maximum number of germ cells? One. Perfectly correct. See, the ovum contains nearly 5 million ova during this period of intrauterine life. Then the number is reduced to 2 million at birth. There is actually a reduction in the number of ova occurs. It becomes 2 million at birth. Further reduction occurs. At puberty, you will be able to see only this question, answer this question, you will come to know. At puberty, the number of ova present in each ovary is about 40,000. So reduction occurs from 2 million to 40,000. Even these 40,000 ova do not undergo full maturation and are released from the ovary during the entire reproductive period of a woman. Only one ovum undergoes full maturation and is released from the ovary during each ovarian cycle. So in that way, if you calculate, only some 400 to 500 ova get a chance to come out of the ovary during the entire reproductive period of a woman. Only 400 to 500 ova come out of the ovary during the entire reproductive period of a woman. From 7 million, 5 to 7 million to 2 million. Before the 7, it can go up to 7 million. I told you only 5, but it can go up to 7 million. To 2 million at birth and 40,000 at puberty. Of the 40,000, only 400 to 500 ova undergo full maturation. Okay. The ovum completes its second meiotic division. What is the major difference between spermatogenesis and oogenesis? In spermatogenesis, from a single cell, primary spermatocyte, 4 spermatozoa are formed. But in oogenesis, from a single primary oocyte, only one ovum is formed. The other 3 cells become polar bodies. They eventually disappear. Only one cell retains all the cytoplasm and that becomes the ovum. So only one cell is formed from oogenesis. This is the difference. Same, all other things are same. First meiosis with a reduction in the number of chromosomes. Second meiosis without further reduction. During first meiosis, the first polar body is released. During second meiosis, the second polar body is released. Here the question is, when does the ovum complete its second meiotic division? Number 4. That means soon after fertilization. You want to choose soon after fertilization. What about first meiosis? When does the first meiosis occur? Just before ovulation? Read this question also. We will see the answer later. Which of the following is a true statement regarding fertilization? Number 3. The spermatozoon penetrates the nucleus of the ovum after entering into its cytoplasm. That's what you think is the right answer for this question. Alright. Which is the usual site of fertilization? Ampulla of the uterine tube. Which is the most dilated part of the uterine tube? Ampulla of the fallopian tube. Which is the common site of tubal pregnancy? Ampulla of the fallopian tube. This is the common site of ectopic pregnancy, tubal pregnancy. It is the usual site of fertilization and this is the most dilated part of fallopian tube. So ampulla, 3 features. Most dilated part, the usual site of fertilization and the usual site of tubal pregnancy. So you have chosen number 3 for this question. The spermatozoon penetrates the nucleus of the ovum after entering the cytoplasm. We will see whether it is true or not. Here you are seeing the ovum released from the ovary is surrounded by several spermatozoa. At what stage of development you see this ovum? First polar body has been extruded and it is at the stage of metaphase where the chromosomes are all arranged in the equatorial region. So under which stage of development the ovum is released from the ovary? Secondary oocyte. So it is the secondary oocyte that is released from the ovary. It has not yet completed its ovulation process, division process, oogenesis. It is the secondary oocyte. The first meiosis in the ovary occurs just before ovulation only. Remember, to form a secondary oocyte. So a secondary oocyte is formed just before ovulation. And it is this cell that is released from the ovary, from the surface of the ovary. It has not yet completed the meiosis. Now it travels along the uterine tube to reach the ampulla where it is going to be surrounded by several spermatozoa, as you see in this picture. And the ovum has a protective covering here. This covering of the ovum, secondary oocyte, is known as zona pellucida. So, the spermatozoa they all try to penetrate the zona pellucida, but only one succeeds. And the spermatozoa gets into the cytoplasm of the ovum. As soon as the spermatozoa enters the cytoplasm, the second meiosis is completed and the second polar body is extruded. So, meiosis is completed only after fertilization by a spermatozoa. If the ovum is not fertilized, the meiosis is not completed and the ovum degenerates within 24 hours. What is the lifespan of ovum in the female genital tract? Less than 24 hours. If it is not fertilized by a spermatozoa within that critical period, even before completing the meiosis, the ovum degenerates. So, second meiosis is possible only after fertilization. But a spermatozoa can stay in the female genital tract. How much time? Period. Viability of the sperm. 48 to 72 hours. 2 to 3 days. 48 to 72 hours. But ovum will degenerate within 24 hours. The secondary oocyte released from the ovary is at the stage of metaphase. First polar body only you are seeing. Now it is going to extrude the second polar body as soon as the spermatozoa goes inside. Let us see that picture now. Yes, a single spermatozoa has gone inside. The acrosomic cap ruptures, the enzyme is released, a small opening is made in the zona pellucida and the spermatozoa goes inside. Now you see the entire spermatozoa inside the ovum, including the tail. Once a single spermatozoa has gone inside, some chemical reaction takes place in the zona pellucida and the opening pore which was made before is now closed forever. The door is shut down. It prevents the entry of all other spermatozoa. So only one spermatozoa can go inside, after which a chemical reaction occurs in that area that prevents the entry of all other spermatozoa. After entering the cytoplasm, the tail degenerates. Only the head undergoes further development. Now you can see here, this is the nucleus of the ovum. This nucleus is called female pronucleus. You can see the second polar body which is now extruded. So the secondary oocyte extrudes the second polar body and its the chromosomes are all condensed to form the female pronucleus. The head of the spermatozoa comes very close to the female pronucleus, but it does not penetrate the nucleus. It enlarges to form the male pronucleus. We shall see that thing in the next picture. This is the second polar body. Here is the spermatozoa, entire spermatozoa inside the cell. Not only the head, but also the tail. Now look at this picture. The tail has now disappeared. The head enlarges to form the male pronucleus and the two pronuclei come closer, lie side by side. This is the female pronucleus. This is the male pronucleus. So there is no penetration of the head of the spermatozoa. Remember, it simply enlarges to form the male pronucleus. Now the chromosome, the nuclear material is condensed and chromosomes appear in these two nuclei, pronuclei. 23 that side, 23 this side. So the diploid number is now retained. 23 plus 23, 46 retained. Now let us see what happens. Second polar bodies. The second polar body, first polar body undergoes one more division to form two polar bodies. In that way, three polar bodies are formed. But by the time the first polar body degenerates, the second one, at a time you will be able to see only two polar bodies. The other one disappears soon. That is why in most of the pictures in the textbooks only two polar bodies are shown. Actually, three polar bodies are formed. But the one degenerates soon. The zona pellucida still persists even after fertilization because its function is like that. What is its function? It prevents the entry of all other spermatozoa. Suppose the zona pellucida disappears immediately after fertilization, other spermatozoa also will get into the cytoplasm. Preventing polyspermy is one of the functions of zona pellucida. What is the other important function? It prevents implantation of the ovum at an abnormal site. How? You see, the fertilized ovum has a property of being stick to any tissue with which it comes in contact. It is sticky, gum-like. Where does the fertilization occur? In the fallopian tube. And suppose the zona pellucida disappears immediately after fertilization, what will happen? Implantation will occur in the ampulla of the uterine tube itself. It should not occur. The zona pellucida should remain until the fertilized ovum reaches the uterine cavity. Only after that it should disappear. If it disappears immediately, implantation will occur there and it will result in tubal pregnancy. To prevent tubal pregnancy, the zona pellucida persists till the ovum reaches the uterine cavity of the uterus. So it does not. So it has two important functions: preventing polyspermy and preventing implantation of the ovum at an abnormal site. Now the two pronuclei come closer and their pronuclear membranes break down, and exchange of genetic material occurs. After that, a zygote is formed. Immediately cleavage begins. As soon as the zygote is formed, division, cleavage occurs. A zygote is formed. Still, immediately metaphase stage has come back. Cleavage spindle. So the ovum completes its second meiotic division soon after fertilization. Suppose you want to choose just before ovulation, what modification will you make in the question? First meiosis. And for this question, you chose number 3. You want to choose the number 3 now. Which is the most right, appropriate? Yes. The infundibulum is not the usual site of fertilization. Spermatozoon does not penetrate the nucleus of the ovum, and the zona pellucida does not disappear soon after the entry of spermatozoa. So the one which we have not chosen has become the right answer for this question. When does the implantation of the fertilized ovum occur? On which day of pregnancy? 4th. That means 6th day of pregnancy. Perfectly correct. By the end of 6th day of pregnancy, fertilization. You can see here the ovum is released, the secondary oocyte is released from the surface of the ovary, and this follicle becomes the Graafian follicle. The Graafian follicle now becomes the corpus luteum after that. Now the ovum reaches the fallopian, the ampulla of the fallopian tube, where it is surrounded by several spermatozoa. Fertilization occurs there, a zygote is formed. It undergoes several multiplication divisions to form a mulberry-shaped mass called morula. Morula, the stage of morula reaches by by the end of third day. Now a small cavity appears in the morula and then thereafter it is called blastocyst. So the ovum, fertilized ovum reaches the uterine cavity on the fourth day. But implantation does not occur immediately, though it has reached the uterine cavity. It remains in the uterine cavity for 2 more days. During this period, some changes occur in the morula. What changes? It starts absorbing the fluid present in the uterine cavity. And the cells are pushed to one side and cells are arranged in 2 groups. Some cells are arranged around the periphery, other cells aggregate and are pushed to one side. These changes occur in the uterine cavity after entering the uterine cavity, that is on the 4th day. Only after 6th day, the ovum, implanted ovum or the blastocyst, now it is called the blastocyst, is implanted into the posterior superior wall of the uterus. So it has to spend at least 2 days in the uterine cavity for some changes to occur in the ovum. 6th day after fertilization, during this period, a blastocyst develops due to accumulation of fluid inside. This fluid is absorbed by the morula from the uterine cavity. And for that change, it has to remain in the uterine cavity for 2 days. On the beginning of the 7th day, the ovum is in contact with the endometrium of the uterus. Once the implantation is completed, the endometrium is no longer called endometrium. What is the term used? Decidua. Yes. There is a block in the isthmus of the uterine tube. This would most likely prevent which of these? It's a very simple question. You have to apply some logic based on what you have studied just now. Number 4, perfect. Because meiosis cannot be completed. If there is a block in the uterine tube, the spermatozoa and the ovum cannot meet. Spermatozoa reaches, ovum is on this side, both of them cannot meet. If they do not meet, meiosis cannot be completed and the secondary oocyte degenerates within 24 hours. So completion of meiosis. Those who do not know anything about this will choose prophase 1 of meiosis, metaphase of meiosis, all those things. But meiosis cannot be complete. Viable congenital malformations that we see in many children due to the action of environmental teratogens, physical agents, chemicals, drugs, infections, ultraviolet radiations, X-rays, all these things, exposure to these agents by the mother is exposed to all these things. They are the environmental teratogens. Rubella infection, for example, can result in congenital malformations. Physical agents, chemicals, drugs, certain drugs. What that tragedy? Do you know the thalidomide tragedy? So, which period of development is the critical period of pregnancy? It is during this period, most of the viable, that means non-lethal congenital malformations occur. It is the period of organogenesis. All organs and organ systems in the embryo develop. If the embryo is exposed to all these agents, environmental teratogens, congenital malformations can occur, viable congenital malformations, during the period of organogenesis. Once all organs and organ systems, development of all these things completed, then even if the embryo is exposed to all teratogens, it will not affect, it will not result in viable congenital malformations, which is that period, the period of organogenesis. Even during this period, even vitamins are not recommended. They also can be teratogenic. No antibiotic, nothing, because of teratogenicity. Development of congenital malformations, which is that period. Number please. Number 3, 4th through 8th week. Perfectly correct. It is during this period from 1st month to 2nd month, completion of the 2nd month, 8 weeks. It is during this period all organs and organ systems develop. Exposure to environmental teratogens during this period can result in several viable congenital malformations. Suppose the embryo is exposed to the environmental teratogens before the period of organogenesis, what will happen? It will end up in spontaneous abortion because viability is not possible. It will end up in spontaneous abortion. Embryo cannot survive if it is exposed to environmental teratogens before the period of organogenesis. Suppose the embryo is exposed to teratogens after the period of organogenesis, what will happen? All organ systems have already developed. A miniature human makes its appearance by the end of 8th week. Thereafter it is called fetus. It is no longer called embryo. Once external appearance and all internal organs, development of all these things completed, now we call that the embryo a fetus. Now environmental teratogens act on the fetus. What will happen? Viable congenital malformations or abortion? No. It will result in intrauterine growth retardation. Intrauterine growth retardation. For example, cigarette smoking by mother in Western culture can result in intrauterine growth retardation after the period of organogenesis. So before the period of organogenesis, abortion. During the period of organogenesis, congenital malformations. After the period, growth retardation only. These are the 4 parts of the uterine tube. The length of the uterine tube is 10 to 12 centimeter and these are the 4 parts. We know the fertilization occurs usually in the ampulla of the uterine tube. Tubal pregnancy. The most common site is the ampulla. Organs and organ systems develop from 1st month to 2nd month, 8 weeks. After that, once the fetus is formed, now growth of the fetus occurs. There is only growth, increase in size of the body. That is happening only after the period of organogenesis. Now, if the fertilization has taken place and implantation also has taken place in the uterine tube, when the fetus grows in size, the tube cannot accommodate. It will rupture, resulting in rupture of tubal pregnancy. So when does the rupture of tubal pregnancy occur? After 2 months. After 2 months. It is during this period the fetus grows in size. That will result in by bleeding sometimes and can result in death of mother. Rupture of tubal pregnancy. But you know, if the fertilization occurs, if the implantation occurs in this part of the uterine tube, the fetus can survive up to 4 months. Up to 4 months survival possible if implantation occurs in this part of the uterine tube. Which part? You may think that the ampulla is the most dilated part, so it can accommodate the fetus for a long period. Which is the narrowest part of the uterine tube? Not even isthmus. Not even isthmus. Look at this diagram. This is the infundibulum. This is the ampulla. This is the isthmus. And also you see a small part of the uterine tube inside the wall of the uterus. This is what is called intramural part. This is the narrowest part of the uterine tube. However, it is a part of wall of the uterus. So if implantation occurs in the intramural part, being a part of the wall of the uterus, it can accommodate the fetus for 4 months. So longest survival is possible if implantation occurs in the intramural part of uterine tube, because it is actually a part of the wall of the uterus, so it can expand greatly. This is an abnormal implantation, abdominal pregnancy. This is implantation in the ovary itself. The spermatozoon reaches the ovary and penetrates the ovary, and implantation occurs even before the ovum is released from the ovary. That results in ovarian pregnancy. And these are the different types of tubal pregnancy. Usually ampulla is the usual site, but it can occur anywhere in the tube. The most common form of ectopic pregnancy is most common type of ectopic pregnancy: abdominal pregnancy, tubal pregnancy, ovarian pregnancy, lower uterine implantation. Tubal pregnancy, yes, 95% of ectopic pregnancy is tubal pregnancy. And also you can see this, it can be implanted in the cervix, lower uterine implantation. These are the different types of ectopic pregnancy, and the one which is common is the tubal pregnancy, that too in the ampulla of the uterine tube. That is why rupture occurs after the second month, after the end of second month, when the fetus grows in size. All organs and organ systems are developed now. And you can see the embryo inside the tube taken. Intramural part is the right answer for this question, not the ampulla which you may think logic-wise. Embryology. Number 4, 3, blastopore. Pore means an opening. Mouth also is an opening. So logic wise, perfect. Do you remember any of this now which you have studied long back? Primitive streak, write short notes on primitive streak, precaudal plate, that much only you know. Let us see how these things appear in the zygote during the latter period of development. a zygote undergoes cleavage, multiplications to form several daughter cells. First, 2 cells are formed, 2 daughter cells, then 2 cells again undergo division to form 4 cells, 8 cells, until a mulberry shaped mass is formed. What is it called? Yes, morula. Usual number of cells in morula 16. From 12 to 32 cells, you can say that a fertilized ovum is morula. You can see the morula now. Zona pellucida is still there. You can see a shell is there covering all the cells. These are the daughter cells formed from the zygote. What is the name given to the daughter cell? Blastomere. The term blastomere is used for all these daughter cells. Blastomere. This is called a morula from 12 to 32 cells. Usually 16, okay, but the range is this, 12 to 32. So it is the morula that enters the uterine cavity on the 4th day. By the time it reaches the isthmus of the uterine, 3 days completed. Then it enters the cavity on the 4th day, remains in the cavity for 2 more days, implantation occurs by the end of 6th day. Okay. Now during this period some changes occur, that is what we are going to see. The zona pellucida is still present. It should not disappear. Now you know why it should not disappear. Now the fluid from the uterine cavity gets into the morula. As a result, the cells are arranged in 2 groups. Some cells are seen around the periphery, other cells aggregate themselves and are pushed to one side. Now we will label. This is what is called an early blastocyst. The outer covering, you can see, the cells which are seen around the periphery are again divided into 2 groups. The outermost cells lose their cell boundary and they form a syncytium. Syncytium means a group of cells without clear-cut cell boundary. So what is the term used for this one? Syncytiotropoblast. The other cells which lie immediately deep to the syncytiotropoblast retain their cell boundaries. Therefore, this layer is called cytotropoblastic layer, syncytiotropoblastic layer. The other cells which are pushed to one side is now known as the inner cell mass, also known as embryoblast, because it is this cell mass that is going to form the body of the fetus, embryo. The tropoblast is associated with, concern with establishment of nutrition to the embryo. It is the inner cell mass that becomes the body, our body. This is the inner cell mass, that is why it is called embryoblast. Tropoblastic layer is concerned with establishment of nutritions to the embryo. How? Several finger-like projections develop from this tropoblast. They penetrate the endometrium of the uterus and also erode the blood vessels in the endometrium of the uterus, and they come in contact with the maternal blood. Now they start absorbing the nutrients in the maternal blood. That is the function of this tropoblast. We can see that now. Here is the blastocystic cavity which is filled with fluid reached from uterine cavity. Now the zona pellucida becomes thinner, gradually starts disappearing, because the embryo has to be implanted, this blastocyst has to be implanted. That is why the zona pellucida now gradually disappears. As long as it is there, implantation cannot occur. Now the zona pellucida has completely disappeared. We see the tropoblast here, outer tropoblast and inner embryoblast. Okay. This is the late blastocyst. It is the blastocyst that gets implanted in the posterior superior wall of the uterine cavity. Blastocystic cavity. Now there is no zona pellucida, ideal time for the blastocyst to get implanted. Implantation 6 days. Now this blastocyst moves towards the posterior superior wall and comes in contact with the uterine endometrium. Now small finger-like projections develop from the trophoblast. These are called chorionic villi. They erode the maternal blood vessels in the endometrium and they come in contact with the maternal blood. Now they start absorbing the nutrients from the maternal blood by the process of osmosis, due to osmosis. You can see after that the entire blastocyst goes inside the endometrium. First only these chorionic villi get into the endometrium and gradually the whole blastocyst goes inside the endometrium. Entire blastocyst, you can see that. Now the cells of the inner cell mass arrange themselves to form a layer, a plate. The cells of the inner cell mass arrange themselves to form a plate, an oval shaped layer. That is called these cells are called hypoblasts. This is the first germ layer to develop, hypoblast, which later becomes the endoderm. Soon other cells also elongate and they also form another layer over this. This is called epiblast. Some cells of the inner cell mass arrange themselves to form a layer called hypoblast. Other cells over this layer elongate and arrange themselves to form another plate called epiblast. The hypoblast is soon surrounded by a cavity. You can see that. What is the name given to this cavity? The hypoblast is soon surrounded by a cavity. You can see that. Yolk sac. Similarly, the epiblast is also surrounded by another cavity. You can see that. Cells migrate from the epiblast from the periphery. They enclose a cavity. Cells of the hypoblast from the periphery also migrate. They also enclose another cavity. This is yolk sac and this is amniotic cavity. So epiblast is related to amniotic cavity. A hypoblast is related to yolk sac. Now the whole thing goes inside and now the structure is completely bathed by maternal blood. Because all blood vessels in that area have been eroded by these projections and blood escapes into the intravillous spaces. Whole thing is now inside. Now the endometrium is no longer called endometrium, now it is called decidua. We use different names for the decidua that we will see later, like decidua basalis, decidua paritalis, decidua capsularis, etc. We shall see them later. Now implantation is completed. Now what happens? This is isolated. The implanted ovum is isolated from the endometrium and we are going to study them in detail. This is now you can label what? Epiblast and this is hypoblast. Epiblast is surrounded by amniotic cavity, hypoblast is surrounded by, yes. It is just like pressing two balloons. When you press two balloons, the pressed area will have two layers. Upper balloon is the amniotic cavity, lower balloon is the yolk sac. And the pressed region is the bilaminar embryonic disc. Because there are two layers now, two laminar, bilaminar embryonic disc. An oval shaped disc, bilaminar embryonic disc with epiblast above, hypoblast below. Two balloons pressed against each other. The pressed region is our body. The pressed region is going to form our body. Embryoblast. So, the embryo is now in the form of a plate, disc, two-layered disc. It was in the form of a mulberry-shaped mass before, morula. Now it is in the form of a two-layered disc. But our body is cylindrical, a tube, trunk, cylindrical with cavities inside. Yes, peritoneal cavity, pericardial cavity and pleural cavity. So, let us see how all these changes take place in this bilaminar disc. Now what happens? The amniotic cavity goes on increasing in size due to accumulation of fluid inside, secretion of fluid inside, amniotic fluid. It gradually surrounds the yolk sac also. As a result, the yolk sac becomes reduced in size. Now this is the amniotic cavity, and you see the yolk sac which is reduced in size now due to enlargement of amniotic cavity. It expands and surrounds the yolk sac also. Eventually the yolk sac disappears. That we will see later. Now the entire embryonic disc is surrounded by amniotic cavity, filled with amniotic fluid. Now embryo is suspended inside the fluid of the amniotic cavity. Now if we examine this bilaminar embryonic disc, we shall see some structures on the epiblast. There is a circular area, this is the epiblast, also known as primary ectoderm. What is the name given to the hypoblast? Primary endoderm. So it is the hypoblast that is formed in the beginning, during the first week. Epiblast is formed during the second week. Which germ layer has not yet developed? Mesoderm. Okay. So this is a bilaminar embryonic disc, isolated and viewed from above after removing this amniotic cavity. It is oval in shape. There is a circular area near the cranial end of the embryonic disc. Cranial end. This circular area is called precaudal plate. Near the caudal end of the embryonic disc, we see a linear elevation. Similarly, there is one more. Near the caudal end of the embryonic disc, we see one more circular area called cloacal membrane. We also see a linear elevation in the caudal part of the embryonic disc. What is the name given to this linear elevation? Primitive streak. This primitive streak presents a small dilatation, a swelling at its cranial end. You can see the swelling here, a slight dilatation which differs from the rest of the primitive streak. This is called primitive node, also known as Henson's node. At the summit of this Henson's node, there is a pit. You can see the pit. What is the name given to this pit? Blastopore. So these are the things that we can observe on the embryonic disc, bilaminar embryonic disc. A linear elevation, a ridge called primitive streak. Its cranial end is expanded to form the primitive node. At the summit of this node, we see a depression called blastopore. We shall take a section of the primitive, this embryonic disc, to see how these things are formed. Precaudal plate is actually formed by elongation of cells of the, this is the epiblast, here is the hypoblast, viewed in a section. And precaudal plate is in fact formed by elongation of cells of the hypoblast in that area. That forms the circular structure when we view from above. This is the primitive node, and you can see the blastopore here, and this is the primitive streak, and cloacal membrane will be seen somewhere here. Okay. The third germ layer, mesoderm, has not yet developed. Now the third germ layer is going to develop from one of these. One of these structures is responsible for the development of third germ layer. Which structure? Anybody? We have seen this precaudal plate, primitive node, blastopore, primitive streak, cloacal membrane. Of the five, which one is going to form or is responsible for the development of third germ layer? Primitive streak. Let us see how it happens. Look at this. The cells of the primitive streak are pluripotent. They proliferate and migrate between the epiblast and hypoblast, as you see in this picture. Cells of the primitive streak proliferate and are pluripotent. They proliferate and migrate from there between the epiblast, spreading between the epiblast and hypoblast, as you see in this picture. Take a section, you can see that. High mitotic activity is seen in the region of the primitive streak. The cells migrate by amoeboid movement themselves. Amoeba-like movement occurs in these cells. They fill the space between the epiblast and hypoblast. They spread between the epiblast and hypoblast. And these cells form the intermediate germ layer, the mesoderm. Afterwards, once the third germ layer is formed, we don't use the term epiblast or hypoblast. We simply call them ectoderm and endoderm. Epiblast now becomes the ectoderm, definitive ectoderm. Hypoblast becomes the definitive endoderm. That we will see now. This is a trilaminar embryonic disc with all three germ layers. This is the ectoderm. Now it is called ectoderm. Previously it was called epiblast. With the development of third germ layer, it is now called ectoderm. Similarly, this is now called endoderm. Hypoblast is now called endoderm. And the middle germ layer, which recently developed from the proliferation of cells from the primitive streak, is now called the mesoderm. Now there are, in this picture you see there are two areas in the embryonic disc, trilaminar embryonic disc, which are not penetrated by the cells of the mesoderm, where the ectoderm and the endoderm are still in contact with each other. One near the cranial end, other near the caudal end. What is the name given to this? Yes, precaudal plate, buccopharyngeal membrane, or the future mouth. There are two areas in the embryonic disc where ectoderm and endoderm are still in contact with each other without the intervention of mesoderm. The cranial one is closed now by a membrane called buccopharyngeal membrane. That is the area where our mouth is going to develop. And this area, in the caudal end also a similar thing is there without any intervention of cells of the mesoderm. Ectoderm and endoderm are still in contact with each other, and this is the cloacal membrane, our future anus. So the beginning point and the end point of our alimentary canal, mouth and anus, are established in these two areas of the embryonic disc. Now cells from the blastopore proliferate here in the midline and extend as a cord. What is it called? Notochord. So notochord develops from the depression blastopore. Cells of the blastopore proliferate to form a cord-like structure exactly in the middle of the embryonic disc. This is called notochord. It is only a temporary structure. It is not found in adult, but this may be found in adult creatures. In some creatures it is found in adult also. But higher animals do not have this thing, because this area determines the central axis of the embryo. It is here the vertebral column develops, but notochord is not concerned with the development of vertebral column. It gives an idea where exactly the vertebral column has to develop in the embryo. As such, it does not form the vertebral column. It determines the central axis of the embryo, the midline of our body. That is the purpose. So this is the mesoderm and this is the endoderm. So we see the notochord in the mesoderm. And how is it formed? By the proliferation of cells in the blastopore. So blastopore is associated with the development of notochord. We call this thing, this area, precaudal plate, because it lies in front of the notochord. That is why we use this term. Pre means before, caudal in front of the notochord plate. Notochord. Same thing, I am going to show that. This is the precaudal plate, our future mouth closed by the buccopharyngeal membrane. This is the future anal orifice closed by the cloacal membrane. Then what happens? Now all three germ layers are formed. Ectoderm, mesoderm and endoderm. All three germ layers are now formed in the embryonic disc. Now what is going to happen in this embryonic disc, that we will see. This disc is going to be folded upon itself in all directions and is going to be converted into a three-layered tube. That is what is going to happen now. Before that, we shall confirm all these structures. Label this, please. Precaudal plate. What is the function of the primitive streak? Yes, the intermediate germ layer, mesoderm, develops from the primitive streak. Because what type of cells does this structure have? Pluripotent cells. Pluripotent, remember. Primitive note. That is why most of the structures in our body develop from mesoderm. Ectoderm gives only the epidermis of our skin. Endoderm becomes the epithelium of the alimentary canal. But all muscles, bone, blood vessels, lymphatic vessels, everything develops from mesoderm. That is why. Because pluripotent. Differentiating into all types of cells. Here is the blastopore. And this plate, this cord is the notochord, and that is why we call this one precaudal plate. It lies in front of the cord. Okay. Now let us see how this embryonic disc is going to be folded upon itself in all directions and is going to be converted into a trilaminar tube. Before that, you see this anomaly. This is a photograph. We see a large tumor in this child. What is it called? Sacrococcygeal teratoma. What is it due to? What is the embryological basis behind the development of such a tumor? Teratoma means a tumor having all types of tissue. Hair, bone, tooth, muscle, everything will be there in the teratoma. After its functional life, the primitive streak completely disappears. After giving origin to the mesoderm, the primitive streak totally disappears. Rarely, in some cases, it may persist near the caudal end of the embryonic disc, where our anal canal, gluteal region, all sorts of things are going to develop. If it is present, because of its pluripotent nature, a tumor can occur in that area. That is what is called sacrococcygeal teratoma, due to the persistence of part of primitive streak, a remnant of primitive streak, sacrococcygeal teratoma. Teratoma, pluripotent cells, primitive streak, pluripotent. Now, the head end of the embryonic disc is folded, tail end of the embryonic disc also is folded. Head fold, tail fold. The amniotic cavity is here, yolk sac is here. Now the amniotic cavity is going to expand, and as a result of which the yolk sac is going to be reduced in size. Let it be. Now sidewise folding also occurs. That is how the disc is converted into a closed tube. Closed cranially, caudally, sidewise, converted into a tube, a cylinder, sidewise folding, sidewise folding. Now it is converted into a three-layered tube. The outermost covering is the ectoderm, the middle layer is the mesoderm, and the inner covering is the endoderm. Now this disc is converted into a tube, closed cranially by the buccopharyngeal membrane, closed caudally by the cloacal membrane. They represent the beginning and the end parts of alimentary canal. Ectoderm becomes the epidermis of our skin, because it is the outer covering of our body, not the entire skin, only epidermis of our skin. The endoderm becomes the alimentary canal. The middle mesoderm contributes to all other structures in our body, pluripotent nature, that is why. Other structures in our body. Look at this, same thing. The limbs develop as outgrowth from the body wall. Near the cranial end, the head develops like this. Caudal end, the gluteal region, buttocks develops. And limbs develop as outgrowth from the body wall. Yes, now you see, the body shape is now attained. But there is no cavity inside in this. But cavities later appear, we will see later. This is how the trilaminar embryonic disc is converted into a cylindrical body. Head end, because of the head folding, head develops. Tail folding, the buttocks develops. Limbs develop as outgrowth from the body wall. Forelimbers, hindlimbers, you can see that. Head area is here. Tail area is here. In higher animals, of course in human beings, tail does not develop, only the buttocks develops. Other animals, tail develops from the tail folding of the embryo. You can see the forelimb bud here. This is going to form the upper limb. This is the hind limb bud. This is going to form the lower limb. Now two swellings you can see here. This is the head swelling, which is going to form the head, and this swelling is the area where the pericardium develops. The heart and pericardium develop. Yes. Now the child will come out of the shell. But we don't have a shell like this. Because of the side folding, lateral folding, the tube, gut is developed. Part of the yolk sac also is absorbed into the gut. That's what we are going to see. For the purpose of our description, our gut, primitive gut, is divided into three parts. We all know. What are the three parts? Foregut, midgut and hindgut. The part of the gut which is in contact with the yolk sac now is called the midgut. Cranial to it is the foregut, caudal to it is the hindgut. This is what happens following folding, sidewise folding. A tube is formed inside, that is our alimentary canal, and is divided into three parts, foregut, midgut and hindgut. The midgut is in open communication with the yolk sac. Now a small diverticulum appears in the wall of the yolk sac. This diverticulum is known as allantoic diverticulum or simply allantois. This diverticulum is responsible for development of urinary bladder in lower animals. But in higher animals, in man, it does not form the urinary bladder. It forms only the apex of the urinary bladder that we will see later. Because of the lateral folding of the embryonic disc, most of the yolk sac is absorbed into the midgut. You can see as a result, it is greatly reduced in size. Previously, it was reduced in size due to the enlargement of amniotic cavity and is converted into secondary yolk sac. That is the primary yolk sac, it develops in the beginning. Due to enlargement of the amniotic fluid, it is reduced in size, and that is what is called secondary yolk sac. Now, because of the lateral folding, due to absorption of the yolk sac into the gut, it is very much reduced. Now, this is the definitive yolk sac. And it communicates with the midgut through a narrow duct. You can see that, narrow duct. Now, the diverticulum, which was actually a small outgrowth from the wall of the yolk sac, has now become a part of the hindgut, because the yolk sac is now absorbed. Now the allantoic diverticulum has now become a part of the gut, following absorption of yolk sac into the gut. This is the definitive yolk sac and communicates with the midgut through a narrow duct called vitello-intestinal duct. Later, both the yolk sac and vitello-intestinal duct disappear. But rarely, the vitello-intestinal duct may persist in some individuals. That can result in what? Meckel's diverticulum, ileal diverticulum. Ileal diverticulum or Meckel's diverticulum is due to the persistence of vitello-intestinal duct. Meckel's diverticulum. Ileal. It is a diverticulum of the ileum. Ileal diverticulum. Now we will see the same thing again. So in the region of the cranial folding, a small pit is formed. due to cranial folding, a small pit develops at the cranial end of the tube. This pit is our future mouth, stomatodium. The term stomatodium is used for this cranial pit and is closed by the buccopharyngeal membrane. Later, the buccopharyngeal membrane disappears and our gut communicates with the exterior through the mouth. Our alimentary canal communicates with the exterior through the mouth, following rupture of buccopharyngeal membrane. The stomatodial region becomes our mouth. Similarly, at the caudal end, another pit is formed. This pit is called what? What is the term used for visualization of rectum and sigmoid colon? Proctoscopy. So this pit is called proctodium and is closed by what structure? Yes, cloacal membrane. Later, this cloacal membrane also ruptures and disappears and anal canal communicates with the exterior. Sometimes the cloacal membrane may fail to rupture. If it happens, a child is born with imperforate anus. Imperforate anus. There won't be any lumen. Because of this, surgical correction is required. That area is cut and removed, and after that the gut will communicate with the exterior. Now you can see here the allantoid diverticulum is a part of the hindgut. So we have two parts for the hindgut. A part cranial to the attachment of the allantoid diverticulum, a part caudal to the attachment of allantoid diverticulum. What is the term used for this part, caudal part of the hindgut? No idea. Okay, that we will see. This is the caudal part of our hindgut. This is the cranial part of our hindgut. Below the attachment of the allantoid diverticulum, it is called the common part. What is the term used for this common area? That is called, okay, this is the proctodium, this is the allantoid diverticulum, and this is called what? We don't have this structure in adult. Cloaca. Cloaca, it is an adult structure in birds. That is, this is the common chamber both for urine and fecal matter in birds. But higher animals, to separate systems develop, carrying the urine, urinary bladder, carrying the fecal matter, alimentary canal, hindgut. So what happens? Some changes have to occur in this area to separate the two systems, urinary system from the alimentary system. This cloaca is going to be separated into two chambers. Let us see how it happens. A small septum extends from the side wall like this, from the junction between the allantois and the hindgut. It goes down, down, down. This septum is called urorectal septum. Very clear. Uro anteriorly, rectum posteriorly. Therefore, the septum which lies between these two parts is called urorectal septum. It goes down and completely, okay, now the cloaca has two parts, a part ventral to the septum, a part dorsal to the septum. The ventral part is called primitive urogenital sinus, and this is what is going to form the urinary bladder. The dorsal part is going to form the rectum and anal canal. This is the primitive rectum. That is why we call this urorectal septum. Now the two things are going to be completely separated. You can see that. Now both are separated. No more cloaca. The ventral part has now become the bladder. Dorsal part has become the rectum and anal canal. The septum grows down and afterwards the cloaca is totally separated into two parts. A ventral part which functions as the urinary bladder, dorsal part of the hindgut, that is the real rectum and anal canal. The allantoid diverticulum forms only the apex of the urinary bladder. And it establishes communication with the umbilicus. What is this structure now called after birth? Urachus. Now it is called urachus. And this structure forms a ligament in adult, exactly in the midline of the anterior abdominal wall, inside. What is it? Median umbilical ligament. Median umbilical ligament. It regresses, no lumen is seen after this. It regresses and forms a ligamentous structure extending from the apex of the urinary bladder to the umbilicus in adult. We all have that median umbilical ligament, remnant of this allantoic diverticulum. No lumen inside. Suppose there is lumen inside, what will happen? If that lumen that will be exposed to the umbilicus and a small hole will be found in the umbilicus. Whenever the person tries to urinate, contracts the bladder, urine can escape through this lumen and come out through the umbilicus. Such a thing is possible. What is this condition called? Urachal fistula or weeping umbilicus. Weeping umbilicus. Umbilicus weeps not with tears but with urine. We will see the anomalies associated with this development of cloaca and the separation later. Now this is the rectum and also the anal canal. You can see the urachal fistula in this case. This is due to the persistence of lumen of the urachus, allantoic diverticulum. So urine escapes through the umbilicus whenever the bladder contracts. Sometimes both ends become ligamentous structures, but the middle part has a lumen, urine collects or fluid collects, and this is what is called urachal cyst. Or urachal sinus also can develop like this. Proximal part closed, distal part patent. These are the anomalies associated with the development of allantoic diverticulum. Urachal fistula, urachal cyst, urachal sinus. Now let us see the anomalies associated with the development of rectum and anal canal. Sometimes the septum does not completely separate the cloaca into a ventral part and a dorsal part. It is incomplete. The separation is incomplete. Now you see bladder communicates with the rectum and anal canal. What is this condition called? Urorectal fistula. Due to persistence of cloaca, not complete, partial persistence of cloaca, should be totally separated into a ventral bladder, dorsal rectum. There are several other anomalies associated with the development of the septum that we will see here in the next picture. Look at this. The urorectal fistula, that is the rectum opens into the urethra or into the bladder. As we have seen before, into the bladder, now it is opening into the urethra. That is possible. The lower part, now look at this one. The rectum in females, the anal canal in females opens into the vagina. Rectovaginal fistula, also another anomaly. See that. Rectovaginal fistula. And the distal part of the anal canal very much regressed. Nothing is there. This is called rectoperineal fistula. And this is the imperforate anus. See the anal membrane, the cloacal membrane persists, proctodium also is there, but there is no lumen. That membrane has not ruptured. Anal membrane has not ruptured, so it results in imperforate anus. So these are the anomalies associated with the development of the caudal part of our gut. So urinary bladder develops from the hindgut. Remember, that means endodermal in origin. Alimentary canal is endodermal. But you see the proctorium, that pit, is from where? Ectoderm. So that is why the lower part of the anal canal is ectodermal in origin, below the pectinate line. It is because of the invagination of the ectoderm forming the proctorium. Upper part of the anal canal, that is above the pectinate line, is a part of the hindgut. Lower part of the anal canal below the pectinate line is a part of ectoderm. That is why the lower part of the anal canal is lined by what epithelium? The epithelium of our skin, stratified squamous epithelium. Similarly, the roof of our mouth is from ectoderm because of the invagination from stomatodium, but floor is from our gut. Roof of the mouth is ectoderm. So, for that way, all orifices in our body are ectodermal in origin. That is why they are all lined by stratified squamous epithelium. Conjunctiva, cornea, external acoustic meatus, nasal orifice, oral cavity, urethral orifice, anal orifice, all orifices are lined by stratified squamous epithelium. That itself is very clear, evident that they are all derived from invagination of our body surface. That means ectoderm. Now, we have spent a lot of time to find out the right answer for this question. This is the basis of our embryology. Once you get this idea, rest of the thing will become simple. Now you know, ectoderm means epidermis of our skin. Endoderm means our alimentary canal. All other structures in our body develop from mesoderm. Very simple. Which one? Yes, precaudal plate. Okay. Which statement about the yolk sac is incorrect? Number 1. We have seen this diagram before. Yolk sac in the beginning is in communication with the midgut. Now with the folding of the embryonic disc, it is reduced in size. It is because it is absorbed into the gut. As a result, the allantois, which was actually a diverticulum of the yolk sac, has now become a diverticulum of hindgut. And what is the term used for the part of the hindgut distal to this attachment? Now cloaca. And it is the cloaca that is partitioned into a ventral part and dorsal part becoming the bladder, dorsal part anal canal and rectum. Clear? Allantois. This is the yolk sac and this duct is the vitello-intestinal duct. Yolk sac is the major site of yolk food for the embryos in lower animals. For example, chick embryo, egg, yolk. That is the only source of food for the chick embryo. But in higher animals, this is not the entire source of food for the higher animals, because soon this function is taken over by placenta. Maternal circulation is connected, linked with fetal circulation. through the development of placenta, and embryo and fetus are nourished by the maternal blood, nutrients from the maternal blood. Since this function is soon taken over by the placenta, yolk is not the major site of storage of yolk in humans, as we see in lower animals. Tea time. But it has an important function in higher animals. You can see some cells developing in the wall of the yolk sac. These are the primordial germ cells, spermatogonia in males, oogonia in females. They actually populate in the wall of the yolk sac. They actually develop from ectoderm. Ectodermal cells are from the primordial germ cells. What do? From the ectodermal area, they migrate into the wall of the yolk sac, where they undergo multiplications. Then what happens? These cells must be there in the gonads. If the gonad is testis, these cells are converted into spermatogonia. If gonad is ovary, the cells are going to be converted into oogonia. So they are now found in the wall of the yolk sac, if not I. Again they have to migrate. So these cells, after populating, after multiplication in the wall of the yolk sac, migrate into the region of the developing gonad and get incorporated into the gonad. So from this you must understand one thing: gonads are not the site of production of primordial germ cells. Spermatogonia are not formed in the testes. Oogonia are not formed in the ovary. Remember, they are actually formed in the ectoderm, populating in the wall of the yolk sac. Then they migrate to the region of the developing gonad and get incorporated within the gonad. So gonads are not the source of primordial germ cells. Remember. Further changes occur in the gonads, that is there, but the source of primordial germ cells not in the gonad, it is in the ectoderm, and multiplication occurs in the wall of the yolk sac. These are the primordial germ cells proliferating in the wall of the yolk sac before migrating into the developing gonad. It contributes to the formation of the gut tube, we have already seen. It is reduced to form the vitellointestinal duct, we have seen. Which is not correct? Exactly. It is going in the opposite direction. Okay. This is not the right one. So we shall have a short tea break now and assemble after 5 to 10 minutes. shall continue. Question number 14. Which of these is not a derivative of forget? Number 3. See, questions like this have been asked previously in most of the entrance exams. Whenever you come across question like this, if you could remember 3 points, we can answer many questions based on the derivatives of the gut and other things. Now we know how our body is formed, the cylindrical body with the outside skin, inside the gut. The first point to be noted, the structures meeting the external environment, the structures meeting the structures of our body or parts of our body meeting the external environment. Which is our external environment? The atmosphere. Meeting the external environment are ectodermal in origin. This is the first point to be noted. Meeting the external environment, atmosphere. That means the epidermis of our entire skin and all body orifices which meet the outer environment are ectodermal, because they develop as invaginations from the body surface, all orifices for that matter. Similarly, point number 2, the parts of our body which meet our internal environment. Which is our internal environment? The lumen of our alimentary canal. The lumen of our alimentary canal is our internal environment. The parts of our body that meet our internal environment are endodermal in origin, because they all develop as diverticula from our alimentary canal. They all develop as diverticula. That is why all of them open into the gut. Those which develop from the endoderm or from the gut will open into the gut. Will open into the gut because they meet the internal environment. Point number 3, the structures which do not meet either the internal environment or the external environment are mesodermal in origin. These are the 3 points to be remembered. Structures meeting the external environment, ectodermal, meeting the internal environment, endodermal, and those which do not meet any of these, either of these, mesodermal in origin. Now, foregut means from which part of our gut in adults is foregut? From the mouth to the opening of the major duodenal papilla in the second part of the duodenum. Up down to that level, it is foregut. Look at this picture. From the oral cavity to the site of opening of the major duodenal papilla in the second part of duodenum, the pink color area. That is foregut. It includes the mouth, pharynx, esophagus, entire stomach and upper half of the duodenum. Then the gut below this level is the midgut. You can see the lower part of the duodenum, jejunum, ileum, appendix, cecum, ascending colon and right 2/3 of transverse colon. Up to the right 2/3 of the transverse colon, midgut. Then the remaining part of our gut, that is left 1/3 of the transverse colon, descending colon, sigmoid colon, rectum and upper part of anal canal. That is why the lower part is not colored. You know why? Yes. Upper part of the anal canal, that is hindgut. That much portion is hindgut. No answer is there for our question. This is how our gut is divided. Which is the artery of the foregut? Celiac trunk. Which is the artery of the midgut? Superior mesenteric artery. Artery of the hindgut? Inferior mesenteric artery. So, each part of the gut is supplied by its own artery. Now, which one you want to eliminate from the list? The respiratory tract is a diverticulum of foregut. That is why the larynx opens into the pharynx, laryngopharynx. Entire respiratory tract, right from the larynx down to the lungs, develops from a diverticulum of the foregut. So, entire respiratory tract from the level of the larynx, endodermal. Biliary tract, the biliary tract opens into the duodenum, into the gut. Therefore, it also should be from the gut. Which part of the gut? Foregut. It is taken along with the foregut. Below that level only midgut begins. Vermiform appendix belongs to midgut. Pancreas, of course, it is a diverticulum of the foregut. Two pancreatic buds develop. Later they fuse to form one pancreas, and the duct of the pancreas, both main and accessory ducts, they open into the duodenum. So they also, these parts, the pancreas also should be definitely from the foregut. Gallbladder is a part of the biliary tract, so it also opens into the foregut. But vermiform appendix, as we see in the picture, is a part of midgut. These are the derivatives of the foregut. Floor of the mouth. Why floor of the, not roof of the mouth? Ectoderm. Why parotid gland is given a chori? Because the duct of the parotid gland opens into the vestibule of the mouth opposite the second molar tooth. That is regarded as a part of roof of the mouth. Therefore, its origin is controversial. If it opens into the floor of the mouth as we see the other salivary glands, submandibular, sublingual, we can say that it is endodermal in origin. But its opening is near the junction between the roof and the floor. Therefore, that chori is there. Nobody is sure whether it is a derivative of the foregut or the ectoderm. All other things are there. Okay, these are the things. The parenchyma of the liver is formed from number 1, 2. Okay, number 1, number 2, then what? Okay, we shall see this picture. At the lower end of the foregut, a small diverticulum appears. That is the beginning of the development of biliary system. This is the hepatic diverticulum. This diverticulum grows into the septum transversum. Before that, it divides into two, pars hepatica and pars cystica. The pars cystica becomes the gallbladder and pars hepatica becomes the parenchyma of the liver. What about the stroma of the liver? Parenchyma means the cellular component of an organ is called parenchyma. Stroma is the connective tissue component of an organ. Yes, you see, the liver is in contact with the diaphragm in adult. Liver is found just below the diaphragm and is in contact. This diaphragm is mainly contributed by septum transversum, which is a fibrous septum developing in the cervical region. Later it migrates down into the region of the trunk and it divides the trunk into upper thorax and lower abdomen. So when it descends into the thorax, it carries its nerve supply also. Which nerve supplies the diaphragm? Now what is its root value? C3, C4, C5, cervical nerves innervating the structure between the thorax and abdomen. This is because the septum transversum migrates from the neck during development. Septum transversum is the main structure that forms the diaphragm. Since it is in contact with the developing lung bud, several fibrous septa extend into the liver parenchyma from the septum transversum. These fibrous septa form the stroma of the liver. So stroma of the liver is contributed by septum transversum. The septum transversum forms the central tendon of the diaphragm. Diaphragm originates from several other sources. The central tendon of the diaphragm develops from septum transversum. It also contributes to the stroma of the liver. Very simple. It's the diverticulum of the foregut, liver and gallbladder. Pancreas also. All salivary glands. These are the parts of the pancreas, adult pancreas. It has a head, neck, body and tail. 4 parts are there. These parts develop from 2 pancreatic buds, dorsal pancreatic bud and ventral pancreatic bud. But one of these is actually derived from the ventral pancreatic bud. Which is that part is our question. Uncinate process. We shall see this diagram. As we discussed before, 2 diverticula appear at the lower end of the foregut in close relation with the hepatic diverticulum. This is the hepatic diverticulum that we have seen already. In close relation with the hepatic diverticulum at the lower end of the foregut itself, 2 buds appear. This bud is placed ventrally. This is the lateral view of the gut. That is why we see this is called ventral. This is the anterior abdominal wall area. This is the posterior abdominal wall area. Okay. So ventral pancreatic bud. This is the dorsal pancreatic bud, which is larger than the ventral. Now what happens during development? The stomach rotates in the clockwise direction. What about the midgut? Midgut rotates in the anti-clockwise direction. Stomach rotates in the clockwise direction, 90-degree rotation only. But midgut rotates 270 degree in the anti-clockwise direction. Those things we will see later. I'm just telling. So when the stomach rotates in the clockwise direction, what happens? The ventral pancreatic bud also rotates along with it due to differential growth. And it comes to lie below the dorsal pancreatic bud. And the 2 buds now are in close contact with each other, therefore they fuse to form a single pancreas. The 2 buds now fuse to form the pancreas. You can see that this is how the pancreas is formed. Now this part of the pancreas is the head, this is the neck, body, tail. But what is this projection of the pancreas called? Uncinate process. Now it is very clear from the diagram. Uncinate process is contributed by ventral pancreatic bud. Uncinate process. Rest of the pancreas is contributed by dorsal pancreatic bud. The pancreas has 2 ducts. A main duct which joins the bile duct to form the hepatopancreatic duct, which opens into the major duodenal papilla. It also has an accessory duct. You can see that. That duct opens into the minor duodenal papilla. Now from the diagram it is clear that the main pancreatic duct is contributed by both the dorsal duct bud and the ventral bud. So main pancreatic duct is contributed by both the ventral and dorsal pancreatic buds. And the accessory pancreatic duct is contributed by dorsal bud only. This is the main pancreatic duct. That is contributed by both the buds. And this is the accessory pancreatic duct contributed only by the dorsal pancreatic bud. So these are the parts of the pancreas developing from the dorsal pancreatic bud except the antennate process which develops from ventral pancreatic bud. In a female child, testes are found. What will be the chromosome sex of the child? That is the question. If you find testes in a female child, what will be the chromosome sex, chromosome constitution, genetic constitution of that individual? Number 1. What is this condition called? Androgen insensitivity syndrome, also known as testicular feminization syndrome. You see, intersex means hermaphroditism. Hermaphroditism is the term used for intersex, ambiguous genitalia. There are two types of hermaphroditism: true hermaphroditism and pseudo hermaphroditism. What is true hermaphroditism? If you could find both ovary and testes in an individual, it is called a true hermaphroditism. Ovitestis, both are found in an individual. On the other hand, if you find gonad of one sex and external genitalia of the opposite sex, it is known as pseudo hermaphroditism. Gonad of one sex, external genitalia, opposite sex. If you find such thing, it is a case of pseudo hermaphroditism. So what about this case? A female child, external genitalia is that of a female, but testis, instead of ovary, testis is found. So this is a case of pseudo hermaphroditism. There are two types of pseudo hermaphroditism: male pseudo hermaphroditism, female pseudo hermaphroditism. What is it? Very simple. If the gonad is testis, it is called male pseudo hermaphroditism. If the gonad is ovary, it is known as female pseudo hermaphroditism. So what is this condition now? It is a case of male pseudo hermaphroditism. Because in a female child, external genitalia female, but testis is found. Based on the nature of the testis, the term male and female hermaphroditism used. Here the testis is found, but it has failed to masculinize that individual. That is why it is called androgen insensitivity syndrome or testicular feminization syndrome. Which chromosome is responsible for the development of testis? Y chromosome, because there is testis determining factor in the Y chromosome. Short limb of Y chromosome, testis determining factor. In the absence of this Y chromosome, the gonad becomes ovary. So in the beginning, during the initial stage of development, the gonad is totally undifferentiated. We do not know whether it is going to become the ovary or testis. It is the Y chromosome that determines. Which part of the Y chromosome? Testis determining factor. As a result, testosterone is secreted. Testosterone is secreted, and this determines whether the gonad should become testis or ovary. In the absence of this TDF, testis determining factor, the gonad becomes ovary. Here the Y chromosome is there, that is why testis has developed. But it cannot bring about a change phenotypically in that individual. It has failed to masculinize that person. That is why this person is a female child. So if you find Y chromosome, then what will be the chromosome constitution of that individual? 46XY. Yes. 46XY. Wherever there is testis, Y chromosome will be there. Without Y chromosome, testis cannot develop. In the absence of Y chromosome, only ovary develops. Any cell in our body can be chosen for karyotyping. But blood can be easily obtained from our body. Therefore, we use the blood cells for doing karyotyping. RBCs cannot be chosen because they are non-nucleated cells, so they won't divide. Only WBCs are chosen. We induce cell division artificially in WBCs which normally do not divide, otherwise. We induce cell division in WBC by adding a substance called mitogen. The commonly used mitogen for routine karyotyping is phytohemagglutinin. Phytohemagglutinin. Once we induce cell division in a culture medium having these cells, mitosis occurs. And we want to study the chromosomes in these cells. The WBC which completes the cell division at the earliest is chosen for karyotyping because we want to give the result to the patient as soon as possible. So the one which completes the cell division early should be chosen for karyotyping. There are 4 WBCs here. Of the 4, which one completes the cell division earlier than all others? That is why we choose that cell. That is lymphocyte. It completes cell division within 3 days. So we can see the chromosomes on the 3rd day itself, beginning of the 3rd day or end of 2nd day after 48 hours. And we can isolate the chromosomes and do the karyotyping. That is why we choose lymphocyte. Others also can be chosen, but it will take time, that's all. We can harvest the chromosomes from the lymphocyte by the end of 3rd day. Human placenta is formed from number 1, do you know what these things are? Chorion frondosum, chorion levae. Of course, decidorbasalis capsularis you might have studied in gynec. But chorion frondosum, chorion levae, we have to see. Look at this picture. This is a diagram of the implanted ovum in the posterior superior wall of the uterus. Soon after implantation, you can see the chorionic villi all around the implanted ovum. The decidua is divided into 3 parts after implantation. This part forms the base of the implanted ovum. That is why this part of the decidua is known as decidua basalis. This part provides a capsule. It covers the implanted ovum. Therefore, it is called decidua capsularis. It provides a covering for the implanted ovum outside. You can see a bulging in the endometrium during this period. A small bulging will be there at the site of implantation. That bulging area, that is decidua capsularis. The rest of the decidua lies outside, therefore called decidua parietalis, also known as decidua vera. These are the three parts of the uterine endometrium, decidua. Now it is known as decidua. Now look at this picture. We have seen chorionic villi all around the implanted ovum in the beginning. Later what happens? The chorionic villi seen in relation to the decidua basalis alone persist. The chorionic villi seen in relation to the decidua capsularis totally disappear. And this part of the chorion, implanted ovum, becomes smooth. That is the reason why we use two terms for the chorionic villi. This part of the chorion is having a rough surface, this part, because of several finger-like projections. That is why it is called chorion frondosum. The other part is very smooth because chorionic villi have disappeared and that part has become very smooth now. It is seen in relation to the capsularis. It is now called chorion leve, smooth chorion. Chorion frondosum means a rough chorion. Now where exactly our placenta develops? It is here. These chorionic villi persist. They erode the maternal blood vessels and come in contact with the maternal blood. What type of placenta is human placenta? Hemochorial. What do you mean by this term? Hemochorial. Mother provides only blood in the development of placenta. Heme. That is why hemo. What does the fetus provide? Chorionic villi extending into the decidua basalis. That is why we call the human placenta hemochorial. Mother's blood, fetus, chorionic villi. So this region is the placenta now. That means decidua basalis is the maternal part of placenta, chorionic villi forming the fetal part of the placenta. This is placenta. Placenta means chorionic villi mixed with maternal blood. On the maternal side you see the cotyledons, fetal side the membranes. So decidua basalis and chorion frondosum form the human placenta. If you examine a chorionic villus, we can see several layers in it. Based on the number of layers, we call them primary, secondary and tertiary villi. The primary and secondary villi are non-functional villi. They are immature, premature villi. Only tertiary villus is fully functional villus. Now try to find out the right answer for this question. We shall see the three types of villi in the diagram later. Number 4, 2. Number 2, number 4. Anybody for number 3? Okay. We will take one villus from here, or some villi. This one villus. This villus has only 2 layers. It is the syncytial trophoblast that extends into the deciduous basalis in the beginning. Later the cytotrophoblast also migrates into it. So this villus is the primary villus having two layers. Outer syncytial trophoblast, inner cytotrophoblast. This is the villus that is formed in the beginning due to extension of the trophoblastic layer of the blastocyst. We have seen already. Now what happens? One more layer is added to this villus. That is, you can see this, one more layer is now added. This becomes the central core of a secondary chorionic villus, that is extra embryonic mesoderm, secondary villus. Now these two are not able to function. What is required for functioning? Fetal vessels, blood vessels should develop in the villus. Only then it can become a fully functional villus. So fetal blood vessels develop in the core of the villus later. This villus is the functional villus. The core of the functional villus, tertiary villus, is the fetal blood vessel. Blood vessels appear, and this villus we call a tertiary villus. Now all these villi are bathed by maternal blood in the intervillus spaces. This villus is bathed by maternal blood all around. And nutrients exchange, gas exchange occur between the blood in the intervillus space and the fetal blood vessels, like this, due by osmosis. All villi are bathed by maternal blood. Gas exchange and nutrient exchange take place between the fetal blood vessels and the blood in the intervillus space. Through these layers. During the last trimester of pregnancy, there is rapid growth of the fetus. It is in great demand for oxygen and nutrients. So, this exchange must be accelerated. But these layers are actually a hindrance. These layers, because all nutrients and oxygen, all those things, pass through these layers. Takes time. So in order to increase the exchange rate, these layers are very much thinned out during the last trimester of pregnancy. Very much thinned out. They do not disappear altogether, but they are very much thinned out. And this side we will see the fetal vessel covered only by a thin layer of these things, and the other side maternal blood. So exchange can take place rapidly. We shall go back to the question. Central core of a secondary chorionic villus is formed by, yes, suppose you want to choose cytotrophoblast, what modification will you make in the question? Primary. Suppose you want to choose fetal blood vessels, tertiary villi. That is how we divide the villi into three parts, three types. Primary, secondary, tertiary. Very simple. Only tertiary villus is functional. Severe vaginal bleeding before parturition. Placenta previa. Perfectly correct. Because in tubal pregnancy, the tubal pregnancy first of all will not last up to parturition. Further, it can produce only concealed bleeding, not vaginal bleeding. Therefore, placenta previa, lower uterine implantation, is the reason for the development of placenta in the lower part of uterus. There are different types of placenta previa, first degree, second degree, third degree, fourth degree, like that. If we happen to see the development of placenta in the lower part of the uterus without covering the internal os, it is a primary, first degree placenta previa. And if the margin of the placenta comes very close to the margin of the internal os, what is it called? Second degree placenta previa. In third degree placenta previa, the placenta covers the internal os. But when the cervix dilates, again the opening becomes patent. So this is third degree placenta previa. Even after full dilatation of the cervix, if the placenta covers the internal os, it is the last degree. So third degree and fourth degree, normal delivery not possible. Oh, normal delivery, vaginal delivery possible, but the mother will die, that's all. Cesarean section must be resorted to. Because the placenta covers the internal os. So it will rupture, resulting in profuse bleeding. So normal delivery should not occur. Somite. Do you remember what this structure? Might have written a short note on somite. Short note on notochord, intraembryonic mesoderm, somite. Only this much you know. Not the story of the development of embryo and fetus. Number 3. Let us see what this somite is first of all, before knowing the developmental period. Yes, now we are very familiar with the parts of the trilaminar embryonic disc. We shall take a section like this, transverse section, to see all 3 layers of the trilaminar embryonic disc. Okay, taken. This is the ectoderm, this is endoderm, and this is middle mesoderm. This middle intraembryonic mesoderm is divisible into 3 parts. This is the notochord, which determines the central axis of the embryonic disc. We see this mesoderm on either side of the midline, either side of the central axis. So what term do you suggest for this mesoderm? Paraxial mesoderm. This one lies between the paraxial mesoderm and the mesoderm which is seen near the periphery. So it is intermediate in position, this mesoderm, therefore called intermediate mesoderm. This one is placed laterally. Lateralmost part of the mesoderm is called lateral plate mesoderm. Like that, we divide the mesoderm into 3 parts. Paraxial, intermediate, and lateral plate are the 3 parts of the intraembryonic mesoderm. Now you see, the paraxial mesoderm produces several swellings on the back of the embryo. The paraxial mesoderm is seen in the form of several swellings on the back, on either side of the future midline. And these swellings are called somites. We shall see them now. So paraxial mesoderm forms the somites. We can see the somites now in this picture. You can see the somites now. Small swellings appear. These swellings begin from the cranial end of the embryo and progress towards the caudal end. Cranio-caudal sequence at the rate of 3 to 4 somites per day. You can see the real embryo, you can see the somites here. The paraxial mesoderm forms these swellings which are found on either side of the future vertebral column. They begin from the cranial end and progress towards the caudal end at the rate of 3 to 4 somites per day. Within 11 days, you can see 44 somites on either side of the midline. 11 into 4, 44 somites appear. When do these somites develop? That is our question. Now we know what somites are. These somites form clear-cut surface elevation on the back of the embryo. We can see them when we do ultrasonography during this period of development. Therefore, this period of development of embryo is known as somite period of development. When is it? That is from 20th day to 30th day of pregnancy. From 20th to 30th day, 11 days are there. So 44 somites appear at the rate of 4 per day from the cranial end to the caudal end of the embryo. And these somites help the physician to determine the approximate age of the embryo. By counting the number of somites present at a particular period of development, we can easily determine the approximate age of the embryo because they are developed at a clear-cut sequence, 3 to 4 per day, like that. You are doing ultrasonography, you are able to count some 24 somites on the back of that embryo. Can you tell me the approximate age of that embryo? For 24 somites to develop, how many days are required? 24 divided by 4, 6 days are required. Somites development occurs from the 20th day. So we add that 6. So 25th to 26th day, approximate age. So we can determine the approximate age of the embryo by simply counting the number of somites present on the back during this period. So somite period is really helpful to determine the age of the embryo. This is one way of assessing the age of the embryo. 20th day to 30th day. Now, what adult structures are contributed by somites? That is our next question. So, it is a mesodermal swelling. Clear? Which mesodermal swelling? Paraxial mesodermal swelling. It is a mesoderm. We know other than the skin, the epidermis of our skin and the mucous membrane of our alimentary canal, all other structures in our body develop from mesoderm, muscles, bones, blood vessels, etc. Now, which one you want to choose from the list? Number 4. Skeletal muscles in our body develop from 3 sources, mainly 2 sources. 1 from the somite, 2 from the branchial arches, pharyngeal arches, 3 from neural crest. Muscles development. Muscles in our body develop from these. Skeletal muscle, only 2 sources, somite and branchial arches. Somites and branchial arches. This neural crest, involuntary muscles, some involuntary muscles of the iris. The muscles derived from somites are supplied by somatic nerves. They are the nerves of our body wall. Nerves of our body wall means all spinal nerves and some cranial nerves. They innervate the muscles derived from somites. And muscles derived from pharyngeal arches are innervated by the nerves of the pharyngeal arches. Nerves of the pharyngeal arches. Which are the nerves of the pharyngeal arches? 5, 7. Nerve of the first arch is trigeminal nerve, mandibular nerve. Second arch is facial nerve. Third arch is glossopharyngeal. Fourth arch is superior laryngeal nerve. Fifth arch disappears soon after development. Therefore, we have only sixth arch, no fifth arch. First, second, third, fourth. After that, sixth only. The nerve of the sixth arch is recurrent laryngeal nerve. They innervate the muscles derived from these arches. Muscles of mastication are derived from first pharyngeal arch. So they are supplied by trigeminal nerve, mandibular nerve. Muscles of the second arch gives rise to the muscles of facial expression. Therefore, they are supplied by facial nerve. There is only one muscle that is derived from the third pharyngeal arch. That is stylopharyngeus, a pharyngeal muscle. That is why this is the only muscle supplied by the glossopharyngeal nerve. Because glossopharyngeal nerve is the nerve of the third arch, so it has to supply a muscle derived from that arch. That muscle is stylopharyngeus. Only one muscle develops from the fourth arch also. This is the only muscle of the larynx that is placed externally. It is the only tensor of the larynx, cricothyroid. So this muscle is supplied by superior laryngeal nerve, the external branch of superior laryngeal nerve. All other muscles of the larynx are derived from sixth arch. Therefore, they are supplied by recurrent laryngeal nerve. Look at this picture. There are some muscles in the head which are not supplied by these nerves but are supplied by other cranial nerves because they are not developing from the pharyngeal arches. Which are they? Which are the muscles in the head which do not develop from pharyngeal arches? That is what we are going to see. Actually, they develop from somites. We will take a single somite and take a section of that somite to see the layers found inside. Now this is the section of a single somite. It consists of three types of tissue. You can see three types of tissue in each somite. We shall name them now. This is the innermost tissue called sclerotome. This is the middle tissue, myotome. This is the outer tissue, dermatome. So each somite has three layers of tissue. Outer layer is known as dermatome, middle layer is known as myotome, inner layer is known as sclerotome. The dermatome gives rise to the dermis of our skin. So dermis of our skin is mesodermal in origin, but epidermis of our skin is ectodermal in origin. Remember, both are from two different sources. The myotome gives rise to all skeletal muscles of trunk and extremities. Trunk and extremities. Muscles of our trunk and extremities, upper limb and lower limb muscles, are all derived from somites. That is why they are supplied by somatic nerves. But some muscles in the head also develop from myotome of somites. Which are they? You will see. You can see these somites which are found in the occipital region, and we concentrate only on the myotome. That is why they are called occipital myotome. What do? They migrate into the region of the developing tongue. The tongue develops in the pharyngeal arches. All parts of the tongue develop from the pharyngeal arches except its muscles. These myotomes from the occipital region migrate into the area, into the pharyngeal arches where the tongue is developing, and they get into the tongue and they form the muscles of the tongue. Muscles of the tongue are derived from occipital somites. That is the reason why the muscles of the tongue are supplied by a cranial nerve which is not a nerve of the pharyngeal arch. That is hypoglossal nerve. Similarly, there are some somites seen in the region of the developing eyeball. You can see that. These somites are called pre-occipital somites. We concentrate only on the myotome. They migrate towards the orbit where the eyeball is developing. They migrate into the orbit and they form all extravocular muscles of the eyeball. Extravocular muscles of the eyeball. That is the reason why muscles of the eyeball are supplied by nerves other than the pharyngeal arches. Which are they? Oculomotor is the main nerve supplying the muscles of the eyeball. Trochlear nerve innervating the superior oblique, and abducent innervating the lateral rectus. So, the oculomotor, trochlear and abducent, these nerves are not the nerves of the pharyngeal arches because they innervate muscles derived from somites. So, in addition to the muscles of the trunk and extremities, two groups of muscles in the head also develop from somites. They are the muscles of the tongue and muscles of the eyeball. All other muscles are derived from somites. Pharyngeal arches here. Now, what about the sclerotome? What does it give rise to? The sclerotome is seen on either side of the notochord cord in the beginning. Later, the notochord cord is replaced by the neural tube. The neural tube gives rise to the brain and the spinal cord. So, notochord cord is replaced and neural tube is there in that position. The sclerotome of one somite is this side, sclerotome of the other somite. They are facing like this, and in the middle neural tube is there. They go towards each other and fuse to form a ring around the neural tube. In other words, a ring around the spinal cord. This ring is the vertebra. The vertebral column develops from the sclerotome, including the ribs, axial skeleton. So, sclerotome gives rise to the vertebral column. Sclerotome of one side, sclerotome of opposite side. They come closer and they enclose the neural tube to form a ring. That ring is our vertebra and the whole thing. In that way, so all these somites are there. So, they form 33 pieces of vertebrae. how many? 33 pieces of vertebrae or 31. 31 pairs of nerve and 33 vertebrae. So, 33 pieces of vertebrae develop from these somites. And what about this thing, our notochord? Does it contribute to any part of the vertebral column? Yes. Yes. The nucleus pulposus of our vertebral column, intervertebral disc, is contributed by the notochord. This is the only adult structure that is contributed by the notochord, and you can include the apical ligament of dens also. Nucleus pulposus of the intervertebral disc and apical ligament of dens, vorontoid process of the second vertebra, cervical vertebra, axis. So, each somite has three types of tissue. Outer tissue becomes the dermis of our skin. The middle myotome forms the muscles of our trunk and extremities and two groups of muscles in the head. Sclerotome forming the vertebral column, including the ribs. Okay. Now we are in a position, muscles of the tongue develop from occipital myotome, and muscles of the eyeball develop from a pre-occipital myotome. That is the reason why, though these muscles are there in the head, are not supplied by the nerves of the pharyngeal arteries. Now the right answer for this question? Which one? Expect all of you to choose the right answer for this question. Definitely, because muscles of facial expression and mastication are developing from pharyngeal arches, branchial arches. Okay. First of all, we should know what neural crust is, how the neural tube develops. In the previous diagrams, we have not seen the development of neural tube. We just saw the folding of the embryonic disc, outer layer becoming the epidermis, middle one becoming all other structures, inner one becoming the alimentary canal. But we didn't see the development of nervous system. Neural crust is a part of neural tube, and it gives rise to several structures in our body. Which one you want to eliminate from the list? 5. Dura mater. Now we shall see the development of neural tube. This is our trilaminar tube with ectoderm outside, endoderm inside, and the middle mesoderm, and this is the area we see the notochord. Okay. Exactly in the midline where we see the notochord, thickening of cells of the ectoderm occurs. A linear elevation, a linear thickening occurs in the region of the notochord, just behind the notochord. This thickening is called neural plate. You can see that. Soon a groove appears in it. A groove has now appeared. Now it is called a neural groove. A groove, wherever it is present, will have two lips. Groove will have a floor and two lips. The name given to the lips of the groove is the neural crust. We give this term neural crust for the lips of this groove, neural groove. Let us see. What happens to this neural groove? It goes on deepening into the mesoderm, as a result of which the two lips come closer and closer and fuse. Soon the tube is detached from the surface. You can see that. It is detached from the surface. Neural groove goes on deepening and the lips come closer and fuse. Along with that, the tube gets detached from the surface. Now it is a tube. Neural crust is the lip. This is the neural tube. That is how the neural tube is formed. So what is the source of the neural tube? Tell me. Ectoderm. Ectoderm. So I told you in the beginning, ectoderm gives rise to only the epidermis of skin. Now you have to modify that statement to this extent. It also gives rise to the entire central nervous system. Entire nervous system. Epidermis. Now we have to use two terms for the ectoderm to avoid confusion. Surface ectoderm and neuroectoderm. Otherwise that confusion will be there. If you use the term surface ectoderm, it is very clear. Surface ectoderm gives rise to the epidermis of our skin. Neuroectoderm gives rise to the nervous system. So that's why we divide the ectoderm into two parts, two groups. Surface ectoderm and neuroectoderm. The lips of the groove. Now in the beginning the two lips are fused and it is a single neural crust. Soon what happens? The lips again separate from each other. They lie on either side of the neural tube, as you see here. This is the neural tube and you can see the right and left neural crust. They lie on either side of the neural tube. So when the neural groove is deepened, and the tube gets detached from the surface, the lips remain separate. And that lip, those lips are the neural crust. They fuse in the beginning to form a single crust. Later again they separate and they lie on either side of the neural tube. What do we see on either side of our spinal cord or the vertebral column? Sympathetic chain, right and left. We see nerve cells in the brain and spinal cord. Do we see neurons outside the nervous system? Do we see neurons outside the brain and spinal cord in our body? Yes. Where? In all ganglia. Ganglia are collections of neurons outside the nervous system. So we see neurons not only within the brain and spinal cord, but also outside. They are seen in a small swellings, ganglia. So what could be the source of these ganglia? Also we see several nerve cells in the wall of our gut. Several neurons are there in the wall of the gut and other organs. Neurons. Failure of development of these nerve cells in the gut result in a disease, especially in the large intestine. Fist-Punk's disease, congenital megacolon. So nerve cells are there outside the nervous system. What will be the source of these nerve cells in our body? What do we see outside the neural tube now? Okay, neural crust. That is going to be the source of all nerve cells, because neural tube can give rise to only the brain and spinal cord. So the source of neurons outside the nervous system could be contributed by the neural crust only. Whatever be the type of ganglia, autonomics, sensory, whatever it is, collection of nerve cells outside means ganglia, that is, neural crust is the source of all these ganglia. Now we shall see. This is again, this one, actually even before folding of the embryonic disc, the neural tube develops. Remember. But just for simplicity, I have shown with after the formation of the tube. Even before the formation of this tube, even in the stage of trilaminar disc, the neural tube develops. Which is the first organ system to develop in our body? Central nervous system. Nervous system is the first organ system to develop in our body to coordinate all the activities of the embryo. So coordination is required. That is why this system develops even before the folding of the embryonic disc. Just for simplicity only I have shown all these things with the tube. Now again the same thing in cross section. You can see the groove here and the lip, the neural crust, and this is the notochord. Now you can see the tube, the groove gets deepened and detached from the surface, converted into a tube. Now the neural crust, lips fused, and again the lips separate. Neural crust. So source of neurons outside the nervous system is from neural crust. Look at this picture. This is the area where we see the neural crust. That is going to form the sympathetic chain. That is why they are seen on either side of the vertebral column or the neural tube. They form the sympathetic trunk, and we see this ganglia, this adrenal gland, adrenal medulla is considered as the largest sympathetic ganglion, where we see the nerve cells. Those nerve cells are contributed by the neural crust, and we see organ plexus, myendric plexus, Auerbach's plexus, like that. So there also we see neurons, organ, also in the wall of the other organs we see nerve cells. They all migrate from the neural crust. So neurons migrate from the neural crust and form the ganglia, the adrenal medulla, the nerve plexus of organs, gut. In addition, neural crust gives rise to the melanoblast of skin color, complexion, melanoblast, pigmented cells of the skin are contributed by neural crust. The orodoblast of our tooth contributed by neural crust. The leptomeninges, it is believed that the leptomeninges are also derived from neural crust. Leptomeninges means pia matter and arachnoid matter. But dura matter is derived from the surrounding mesoderm, not from the neural crust. The muscles of the iris, sphincter pupillae, ciliaris, also are derived from neural crust. Several muscular and skeletal elements of the head and neck are also said to be derived from neural crust. Muscular and skeletal elements of head and neck. Even it plays a vital role in the development of heart also. The AV cushion, atrioventricular cushion that we are going to see during the development of cardiovascular system, also contributed by the neural crust. So this plays a very vital role in the development of several structures in our body. Which one? Yes. So those who do not know will choose one of these ganglia. Oh, there are 3 types of ganglia, we can choose one. Could be. Ganglia means outside neurons, therefore source is from neural crust only. Which one? I expect all of you to choose the right answer for this question. All of the above. Corpus lucidum provides the epidermis. Mesoderm, that means the dermatome of the somite, that provides the dermis. Neural crust provides the pigmented cells of the skin. So, skin is a very broad term in embryology. Derived from 3 sources: ectoderm, mesoderm and neural crust. Whenever you read a pedigree chart, what all things you should note to find out the mode of inheritance in that family. The patient who comes to you with a genetic disorder is called a proband. Males are represented with a square, females are represented with a circle. If shaded, they are the affected individuals. Whenever a patient comes to you with a genetic disorder, we have to trace at least 3 to 4 families, if possible, to find out the mode of inheritance of that particular disorder in that family. Here in this picture, in this pedigree chart, 3 generations have been traced. First generation, second generation, third generation. 3 generations have been traced. Shared individuals are affected individuals in each generation. What all points to be noted in a pedigree chart to find out the mode of inheritance? 4 modes of inheritance are there: autosomal dominant, autosomal recessive, sex-linked dominant, sex-linked recessive are the 4 modes of inheritance. How will you arrive at your diagnosis? Any idea about this one? X-linked dominant. Okay. 2. autosomal recessive, then what? Others have no idea, and those have got some idea have chosen X-linked dominant, autosomal recessive, then what else? Autosomal dominant, okay, has come now. Only one thing remains now. X-linked recessive. Now the points to be noted. Okay, read this pedigree chart also. Another pedigree chart is given. In this chart, 4 generations have been traced. First generation, second generation, third generation, fourth generation. What is the mode of inheritance in this pedigree chart? Number 2, again autosomal recessive. All right. Okay, we will see the first question now. What are the points to be noted? First point, most important. See whether the disorder is found in all generations without skipping any generation. What about this pedigree chart? So this is more in favor of a dominant disorder. Only a dominant disorder will manifest in all generations without skipping any. This is the point, first point to be noted. Very obvious from the pedigree chart, it is a dominant disorder. Point number 2, see whether males and females are affected in equal proportion. Affected? Both males and females are affected? Okay. Keep that thing. Now, I am going to add one more point. See whether the male members, affected male members in the family have transmitted the disease to their sons. Whether the affected male has transmitted the disease to his son. Yes or no? So if a male transmits the disease to his son, the defect must be, so we have taken 3 points. Without skipping any generation, the disorder is found in all individual generations. That is more in favor of an autosomal dominant disorder. There are 2 types of dominant disorder, autosomal and sex-linked. Now we want to differentiate whether it is autosomal dominant disorder or sex-linked dominant disorder. Here the father has transmitted the disease to his son in this one. In this one, it is also a dominant disorder because, yes, but males and females are not equally affected here. Females are more in number. Now the father has transmitted the disease only to his daughters. None of his sons is affected. He has not transmitted the disease to any of his sons. This is the major difference between the 2 charts projected. In this one, father has transmitted the disease to his son. In this one, he has not transmitted the disease to his sons. What do you understand from this? Suppose the defect is in the X chromosome. it be. X chromosome defect, X-linked dominant disorder. If the defect is found in the autosome, it is autosomal dominant disorder. Let it be. Suppose the defect is there in the X chromosome. Father can contribute only Y chromosome to his son. If he contributes X chromosome, the person is not son, daughter. So father cannot transmit the disease to his sons if the defect is found in the X chromosome. On the other hand, if the defect is found in the autosome, father can transmit the disease to his sons. Very simple. Now tell me, what is the mode of inheritance in this chart? Father has transmitted the disease to his son. So it is a case of autosomal dominant disorder. In this case, father has not transmitted the disease to any of his sons. Therefore, the defect must be in X chromosome. So what is the mode of inheritance here? Yes, we will see. These are the points to be noted whenever you read a pedigree chart. Multiple generations affected dominant disorder. Males and females are affected in equal proportion. Male to male transmission does occur. That point must be underlined. Only in autosomal disorder. Male cannot transmit the disease to his son in sex-linked disorder. Even in your explanatory note, take down. Everything is there in your explanatory note. So, for this chart, your diagnosis, mode of inheritance is autosomal dominant because male has transmitted the disease to his son, other male. But in this case, only daughters are affected because he contributes the defective X chromosome to his daughter. One normal chromosome from the mother and defective X chromosome from the father. That is why all daughters are affected. He contributes X chromosome, defective X chromosome to his daughters. Therefore, it is a case of X-linked dominant disorder. So, how will you find out whether it is a dominant disorder or not, irrespective of whether it is autosomal or sex-linked? Without skipping any generation, all generations will be affected. That is the point in favor of a dominant disorder. And for differentiating this into whether it is sex-linked or the autosomal dominant, see whether the affected male has transmitted the disease to his son or not. If he has transmitted the disease to his son, it is a case of autosomal dominant. Not transmitted means X-linked dominant. Because sex-linked dominant disorder, sex-linked means X-linked only. These two terms are synonymous. Because most of the defective genes are found only in the X chromosome, not in the Y chromosome. Y chromosome is, hairy pinna, some congenital baldness. These are the things associated with Y chromosome. Most of the sex-linked disorders are due to defective genes in the X chromosome. That is why these two terms are used synonymously. X-linked and sex-linked are one and the same. X-linked and sex-linked means one and the same. We don't have Y-linked disorder. So based on what we have studied so far, find out the answer for this question. I have given you the clue also before with 3 points. Four, respiratory tract communicates with the gut. Hepatobiliary tract communicates with the gut. Ducts and acini, of course, they are in communication with the gut. So they must be endodermal in origin. But spleen does not communicate with the gut or communicate with the body part surface. Therefore, must be mesodermal. This is, whenever you come across questions like this, see whether that part communicates with the gut or with the surface of our body. Now, another exercise. Now, you are all trying to make them communicate either with the gut or with the surface, but some of them do not fit. But we cannot modify our statements. So when you come across a question like this, then what will you do? One thing you can eliminate immediately, pancreas, definitely a part of endoderm, but other three, what will you do? Thyroid gland and parathyroid, do they communicate with the gut or at least with the outer surface? No. Thymus, does it communicate with the gut or outer surface? That also. So only one of these is the right answer for our question. We cannot choose many. What is the development of thyroid gland? It develops from the thyroglossal duct. Where does this duct originate from? From the foramen cecum of the tongue, a part of our gut. Therefore, it was in open communication with the gut in the beginning during development. Later, the proximal part of the upper part of the thyroglossal duct disappeared, only the distal part enlarged and became the thyroid gland. So, what is the source of, what is the germ layer associated with the development of thyroid gland? Endoderm. Parathyroid and thymus, all these things develop in the wall of the pharynx, pharyngeal pouches. Pharyngeal pouches are part of the pharynx. Pharynx is a part of our gut. So they are also from, those details we will study later. Development of parathyroid, thymus, tonsil, palatine tonsil, thymus and parathyroid. They all develop from pharyngeal pouches. So they are also endodermal in origin. Adrenal cortex, no connection with the internal environment or with the external environment. Therefore, it must be mesodermal in origin. Urogenital system, definitely from mesoderm, no doubt. Anything other than the epidermis of our skin and nervous system, then the gut, epithelium of our gut, should be from mesoderm only, no doubt. But which mesoderm is our question? We have so far studied only three. Now one more is there. What to do? We have seen already paraxial, intermediate, lateral plate. Now one more has been added, which we haven't seen before. So what to do? We will see, we will find out how that one also comes. Any idea about the development of urogenital system? Anything you want to choose? Gus. 2. Intermediate, mesoderm. Lateral plate. Okay, no problem. Paraxial you won't choose because we have seen all three. Dermatome, myotome and sclerotome and the derivatives of all these three. So you won't choose paraxial mesoderm. It could be either intermediate, lateral plate or somatopleuric. Soma means body wall. Urogenital system has nothing to do with our body wall. Outer surface of our body. Therefore unlikely. Okay, we will see. You remember having seen this diagram before with the three mesoderm? Okay, now we will see the diagram again. You see in this picture, the lateral plate mesoderm here. You see a small opening, a cavity appearing in the lateral plate mesoderm. This cavity soon enlarges to form a very large cavity like this. This is a cavity formed inside the embryo, embryonic plate, embryonic disc. Therefore, what is this cavity called? Intraembryonic coelom. A cavity appears in the lateral plate mesoderm, intra-embryonic coelom. It is a closed cavity now. What happens later? It opens outside laterally. Later, it communicates with the exterior. As a result, the lateral plate mesoderm has now disappeared. Now in that place we see two types of mesoderm. One in contact with the ectoderm, other in contact with the endoderm. So we have to give two names for the mesoderm which are newly formed from the lateral plate mesoderm. This one is in contact with the ectoderm, therefore called somatochloric mesoderm. Because soma means body wall, ectoderm is going to form the body wall. That is why. This one is called splangnochloric mesoderm. Splagnology means study of gut. So this one is going to be associated with our gut, musculature of the gut. That is why splangnochloric mesoderm, splagnology, viscera. So in the beginning a small opening is found in the lateral plate mesoderm. Later it increased in size and formed some actual cavity called intraembryonic coelom. Then it opens outside now. You can see this cavity resembling a horseshoe when viewed from above. You can see the cavity. This horseshoe shaped cavity is the intraembryonic coelom, and it is here it opens. Intraembryonic coelom. Otherwise, our body and the body of the earthworm will be similar. It will become similar because earthworm does not have any cavity inside. It has only three germ layers: outer ectoderm, middle mesoderm, inner endoderm. No cavities in between, but we have cavities. Our body surface is separated from the gut by a cavity. That is our peritoneum, peritoneal cavity. So a cavity has to appear inside our body to separate the viscera from our surface, from the body surface. That is why such a cavity is formed. We will see this in detail. Now we will take a section like this. This is the situation. We have seen this diagram before. Only after the formation of a cavity, the disc is going to be folded on all sides. But I showed you even before, just for understanding purpose. Actually, it is now the disc is going to be folded after the formation of the intraembryonic coelom. Let us see if it lateral folding I am going to show, sidewise folding. Yes, progressing. Yes. Yes. Now this is the thing. Sideways folding occurs after the formation of a cavity in the lateral plate mesoderm. Now we see a trilaminar tube with a cavity inside. This is our paraxial mesoderm. This is the intermediate mesoderm. Now there is no lateral plate mesoderm. In its place we see one mesoderm which has gone along with the ectoderm, that is somatochloric. The other mesoderm which has gone along with the endoderm. This is our gut. It is surrounded by which mesoderm now? Splagnolochloric mesoderm. This splagnolochloric mesoderm forms the musculature of our gut, smooth muscle of our gut. It is because of this muscle, intestinal colic. You get inner circular layer, outer longitudinal layer. You might have studied histology of the gut. coelom is inside the tube and is going to form the three body cavities: pericardial cavity, pleural cavity, and peritoneal cavity. We will see. This mesoderm, what is this mesoderm? Paraxial mesoderm. What is the other name given to it? Somite. What are the three parts of the somite? Yes. Dermatome, scleromyotome, and sclerotome. What does the dermatome give rise to? Dermis. Myotome, muscles of trunk and extremities, plus two muscles in the head. What about sclerotome? Axial skeleton. Okay. Vertebral column and ribs. Now what about the intermediate mesoderm? This is the mesoderm that forms most of our urogenital system. Intermediate mesoderm forming the urogenital system. What is this mesoderm called? Somatopleuric mesoderm. It gives rise to the appendicular skeleton, the skeleton of our limbs, and the subcutaneous tissue and outer layer of body cavities. Because it lies next to the skin, therefore subcutaneous tissue, naturally. And the outer layer of body cavities means the parietal peritoneum, parietal pericardium, and parietal pleura. Outer layers. That is why all these things are innervated by the nerves of the body wall. Therefore, they are all sensitive to pain. All sensitive to pain. Pericarditis is painful. Pleuritis is painful. Peritonitis is painful. Inflammation of the outer layer of our body cavities. In the outer layer is innervated by the same nerves innervating the body wall. Intercostal nerves. Same intercostal nerves innervate the abdominal wall also. Lower 6 intercostal nerves innervating the abdominal wall. So all are painful. Parietal peritoneum is innervated by the same nerves. And parietal pleura is innervated by the intercostal nerves. That is why pain. And the fibrous pericardium and the parietal layer of the serous pericardium are innervated by the phrenic nerve, which is a somatic nerve innervating the diaphragm. So same nerve innervates these structures also. That is why pericarditis is painful. But you see the splanchnopleuric mesoderm, it forms the musculature of the gut and is innervated by autonomic nervous system. Therefore, not sensitive to pain. Structures innervated by autonomic nervous system are not sensitive to external sensation, especially the pain. But structures innervated by the nerves of the body wall are sensitive to external stimulation, sensitive to pain. So we have already completed the structures derived from the paraxial mesoderm. And regarding the lateral plate mesoderm, not much complicated, dividing into two parts, somatopleuric, splanchnopleuric. Splanchnopleuric becomes the musculature of our gut. And somatopleuric forming the appendicular skeleton, subcutaneous tissue, outer layer of the body cavities. Over. So we are going to study the intermediate mesoderm in detail. With that, derivatives of the mesoderm also will be over. Major part of embryology. Now you see, when the lateral folding progresses, you see, as you see here, lateral folding, they come closer anteriorly and fuse here. Now the gut is suspended by two mesentery. One from the posterior abdominal wall, dorsal mesentery. Other one from the anterior abdominal wall, that is ventral mesentery. If the two mesenteries persist, we will have two peritoneal cavities. right peritoneal cavity and left peritoneal cavity. Now the peritoneal cavity is divided into two cavities, right cavity, left cavity. But this should not occur. Therefore, which mesentery should disappear to make it one? Yes, ventral mesentery disappears and the gut is suspended only from the posterior abdominal wall. And this is the dorsal mesentery. Now we need not use this adjective unnecessarily because we have only one mesentery. So there is no point in using dorsal mesentery. We simply call it mesentery. That's all. The site of fusion of the two edges of the embryonic disc is seen in the midline in the anterior abdominal wall. What is it that called? Linea alba. The linea alba that we see in the anterior abdominal wall shows the site fusion of the two edges of the embryonic disc. That extends into the scrotum also. You can see the median raphe in the scrotum. That median raphe also is the site of fusion in males. Such a fusion does not occur in females, so vulva and vagina, all those things develop in that area, that's all. So this now becomes a single peritoneal cavity. This one is the parietal peritoneum, that is somatopleuric mesoderm. This peritoneum, visceral peritoneum, that is splagmopleuric mesoderm. Parietal peritoneum, somatopleuric, visceral peritoneum, splagmopleuric. Okay. Now you see, ventral mesentery does not disappear altogether. In some areas, ventral mesentery can be found. One such thing is falciform ligament. It extends between the liver and the anterior abdominal wall. This falciform ligament is a remnant of, a derivative of ventral mesentery. Not only this one, we see one more. You can see that after covering the liver, that mesentery extends into the stomach, into the lesser curvature of the stomach. What is this fold? Lesser omentum. So falciform ligament and lesser omentum represent the ventral mesentery. So liver bud, the hepatic diverticulum grows into the ventral mesentery. That is why it is covered throughout by the ventral mesentery. That becomes the covering, the intraperitoneal organ, covering of the liver. And part between the liver and the anterior abdominal wall becomes the falciform ligament, and the part between the liver and stomach becomes the lesser omentum. That's all. So questions can be said, which of these represents the ventral mesentery of the embryo during embryonic period? Okay, 1, 2, 3, 4 given. So you should know two things, falciform ligament and lesser omentum. We can see this yolk sac still connected to the midgut through the vitellointestinal duct. Later, vitellointestinal duct and yolk sac disappear completely. This now becomes the gut, and this is what is called now simply mesentery. Simply mesentery, and this is the gut, and this is the peritoneal cavity. Here is the peritoneal cavity. Now a 3-layer embryonic tube with a cavity inside. That is the difference. That is why gut is totally separated from the body wall. Otherwise, it was in communication with the mesoderm before. Now it is totally separated. A cavity has developed inside. So we all come under coelenterata. Animals with coelom inside, cavity, coelenterata. Earthworm, no coelom inside. shown you with symbol diagram how the peritoneal cavity develops. But what about the pericardial and pleural cavities? Pleural cavities are 2 in number. How do they develop? That is what we are going to see now. This is the region where the heart is going to develop. So that part of the coelom becomes the pericardial cavity. And these 2 areas on either side, they become the pleural cavities. The lateral folding of the embryonic disc is more marked ventrally, and where we see the 2 peritoneal cavities fuse to form a single peritoneal cavity. Then you may ask, all these 3 cavities are now communicating with one another, but in adult all are independent cavities. How do these things develop? How do these things occur later? How they are separated? That is what we are going to see in the next picture. Membranes appear between these to separate these cavities. You can see this is the pericardial cavity. It is going to be separated from the pleural cavity by a membrane. This membrane is called pleuropericardial membrane. So membranes appear between these cavities and separate them into 3. How? You see a membrane here between the pericardial cavity and pleural cavity. This is pleuropericardial membrane. Similarly, the pleural cavity is separated from the pericardial cavity by another membrane. That is called the pleuroperitoneal membrane. This membrane also forms a part of the diaphragm. If diaphragm does not develop, then the abdominal cavity, peritoneal cavity will communicate with the pleural cavity. But pleural cavity is totally separated from the peritoneal cavity with the development of diaphragm between. So, pleuroperitoneal membrane also contributes to the development of diaphragm. Not only septum transversum. And the 2 pleural pericardial cavities, because of the lateral folding of the embryonic disc, come closer and fuse to form a single peritoneal cavity. And lungs develop here, right lung, left lung. Each one invaginates the pleural cavity from the medial side, thus converting the pleural cavity into 2 pleura, visceral pleura and parietal pleura, by invaginating into the pleural cavity. Heart develops here. It invaginates the pericardial cavity from behind and converting the pericardial cavity into an outer pericardium and inner pericardium. Fibrous pericardium and parietal pericardium outside, visceral pericardium inside. And the fluid between these 2 is the pericardial fluid. So visceral pericardium is nothing but the epicardium of the heart. Visceral layer of the pericardium is the epicardium. Heart wall has 3 layers: inner endocardium, middle myocardium, outer epicardium. Outer epicardium is nothing but the visceral pericardium. That is this pericardial cavity inner layer. So, urogenital system develops from intermediate mesoderm. We shall discuss one more questions and we shall have lunch break after this. With same thing, urogenital system, some more details. We will complete this system. Gonads develop, we know, developing from intermediate mesoderm. But which part? These are the parts of the intermediate mesoderm that we are going to see now. From this type of question has been asked repeatedly in your exams. From what? 1. Look at this picture. Gut is there, mesentery is there, peritoneal cavity is there, You can see the neural tube flanked on either side by the neural crust. The degenerating notochord cord is seen here, and this is the region where we see the intermediate mesoderm. This is the intermediate mesoderm. What do we see in this intermediate mesoderm? What happens to this intermediate mesoderm later? From the picture, it is very clear that it presents two swellings. Intermediate mesoderm presents two swellings, a medial swelling and a lateral swelling. So we have to give two names. The medial swelling is known as genital ridge, also known as gonadal ridge. What about the lateral swelling? The lateral swelling is known as nephrogenic cord, urogenital, nephrogenic cord. In relation to the nephrogenic cord, two duct systems develop. You can see a large duct in relation to the nephrogenic cord. One of these is quite large, and this duct is called mesonephric duct, also known as Wolffian duct. Lateral to this mesonephric duct, one more duct appears here. This duct lies lateral to the mesonephric, therefore called what? Paramesonephric duct, also known as Mullerian duct. These are the four parts of intermediate mesoderm. Two swellings and two ducts. Two swellings, medial swelling, gonadal ridge, lateral swelling, nephrogenic cord. And the two duct systems developing in relation to the nephrogenic cord are the mesonephric duct and paramesonephric duct. So all these things together known as intermediate mesoderm, from which urogenital system develops. Urogenital. Most of the urogenital. Why I am using most of the urogenital? Because we have already seen the development of urinary bladder, which is a part of the urinary system. Is it from the intermediate mesoderm? No, it is from the hindgut. That is why most of the urogenital system. Now, the gonadal ridge becomes the gonad, ovary or testis. Nephrogenic cord, as the name suggests, forms what? Kidney. Excretory part. Nephrons of the kidney develop from, you can see that several nephrons appearing in the nephrogenic cord. You can see that small tubules appear. They all form the nephrons of the kidney. The paramesonephric duct gives rise to the female reproductive system. Female reproductive system means uterus and uterine tube. What about mesonephric duct? It gives rise to the parts of male reproductive system. This is the summary of intermediate mesoderm. So we see four parts in this mesoderm, two swellings and two ducts, medial swelling, gonadal ridge, gonad, lateral swelling, kidney, paramesonephric duct, uterus and the uterine tube, mesonephric duct, parts of male reproductive system. Summary. Now we know the answer for this question. Genital ridge. One more. We have two parts for the kidney. Nephrons of the kidney, that is the excretory part. The collecting part of the kidney means the minor and major calyces. the pelvis of the ureter, then the ureter goes down. So they form the collecting part of the kidney. Nephrons form the excretory part of the kidney. Collecting part of the kidney develops from, we have seen already, the nephrogenic cord giving rise to nephrons, excretory part. And what about the collecting part of the kidney, minor and major calyces, right from the collecting tubule, then the minor calyces, major calyces. Where do they develop from? Now you are exposed to some more terms, pronephros, mesonephros, metanephros. So far we have seen only nephrogenic cord. I told you only the medial swelling, gonadal rit, lateral swelling, nephrogenic cord. Now some more terms have been added in the intermediate mesoderm. What to do? We shall see them now. This nephrogenic cord has three swellings. Each swelling becomes a kidney. Pronephros, mesonephros and metanephros are the parts of nephrogenic cord. This is the medial swelling. We know the name of the medial swelling. What is it? Genital ridge. Okay. So nephrogenic cord is seen in the form of three swellings. Pronephros, mesonephros, metanephros. One on either side. So 6 swellings are there on both sides. 3 on one side. Therefore 6 kidneys develop in the beginning. Each one becomes a kidney. But in higher animals we see only 2 kidneys. One on either side. But in lower animals all 6 kidneys persist. So which of these persist to form the kidney in higher animals? And which nephros disappear later? Look at this picture. One side only shown. Pronephros disappears. Mesonephros also disappears. Only metanephros persists. That metanephros becomes the excretory part of kidney. We have already seen the development of a duct within the nephrogenic cord. What name did we give? Yes, this is the duct. Why that duct should be called mesonephric duct? This is because this duct develops in relation to the mesonephros. Though the mesonephros disappears, the duct in that area persists. That is why we call this duct mesonephric duct. Because it is seen in relation to the mesonephros. As we have discussed before, mesonephric duct is associated with the development of parts of the male reproductive system. But you see, irrespective of the sex, it gives rise to a diverticulum. You can see that from the picture. This diverticulum is called ureteric bud or ureteric diverticulum. This diverticulum grows towards the metanephros and fuses with it. It grows towards the direction of the metanephros and fuses with the metanephros. It gives rise to these two things form the kidney. How? Metanephros forming the nephrons of the kidney and the ureteric bud gives rise to the collecting part of the kidney. So collecting part of the kidney, including the ureter and also the trigone of the bladder, are developing from the ureteric bud. Ureter and the collecting part of the kidney. The two things come closer and fuse to form the whole kidney. The rest of the ureteric diverticulum becomes the ureter. Suppose the ureteric bud fails to fuse with the metanephros, what will happen? What anomaly can you expect? You will have a kidney without ureter. If you apply logic, you will have a kidney with nephrons only, and ureter will be there separate, totally separate, free from communicating with the kidney. This is what we normally expect. But in reality, this does not occur. The kidney does not develop. It results in agenesis of kidney. If the two things do not fuse, kidney will not develop. So these two things have signed an agreement. Only if you come and join me, we can function together. Otherwise, we will commit suicide. Okay, now the ureteric bed has gone inside the metanephros, good, but it fails to communicate with the nephrons. What will happen? See, nephron means proximal starting from the Bowman's capsule, then convoluted tubule, loop of Henle, distal convoluted tubule, then the collecting tubule. So collecting tubule onwards from the ureteric bud up to the distal convoluted tubule, nephron. That is from the metanephros. If the two things do not communicate, what will happen? The collecting tubule should communicate with the distal convoluted tubule, only then urine can flow through. If they do not communicate, what will happen? This one has come closer, nephron, distal convoluted tubule. This ureteric bud that branches, collecting tubule has there, but there is no connection between them. It will result in polycystic kidney, congenital polycystic kidney, because urine formed in the nephron cannot flow and all nephrons are dilated, resulting in polycystic kidney. So communication must be established. Simple entry is not sufficient. Entry into the metanephros is not sufficient. They should establish communication, connection. Only then normal kidney development is possible. If they do not communicate, congenital polycystic kidney will occur due to dilatation of all nephrons by the urine formed in the nephrons. So failure of union will result in agenesis of kidney, and failure of communication will result in polycystic kidney. Yes. So collecting part of the kidney develops from, now you know the answer for this. What about excretory part of kidney? Metanephros. The term, specific term used for the metanephros is metanephric blastema. Suppose you come across such a term as one of the choices, you should not wonder. Metanephric blastema. Is it different from metanephros? Okay, now one more question is there about the kidney. With that, I think it will be over. Polyhydramnios, excessive amniotic fluid in the amniotic cavity. What could be the reason? The other condition is oligohydramnios. Which of these is likely to produce polyhydramnios? Number 3, duodenal or esophageal atresia. You see, the kidneys start functioning during intrauterine life itself. Urine is formed and is added to the amniotic fluid. The main contribution of amniotic fluid is fetal urine. fetal urine is the main source of amniotic fluid, and urine is excreted by the fetus every day. And if it is not drained properly, what will happen? The quantity of the amniotic fluid will definitely increase. How is it eliminated? The fetus itself swallows the amniotic fluid. Several milliliters of amniotic fluid swallowed by the fetus and is absorbed from the alimentary tract and is available for recirculation. That is how the excess fluid, amniotic fluid, is removed from the cavity. For that, which system should function normally? Gastrointestinal tract must develop properly. Only then the swallowed fluid can be absorbed by the fetus. If there is any defective development of the alimentary canal, this is not possible. So, the quantity will go on increasing. That will result in polyhydramnios, that is duodenal atresia, non-development of duodenum or non-development of esophagus. The fetus cannot swallow the fluid, or even if it swallows, cannot go inside, it will come out. The other condition can occur, oligohydramnios, can be due to, yes, urine is not added to the amniotic fluid. That means non-development of kidney, or kidney and everything has developed, but there is some obstruction in the urinary tract. Obstructive uropathy. These two things can produce oligohydramnios. Bilateral agenesis of kidneys and obstructive uropathy, some obstruction in the urinary tract. Urine is not added, so quantity of the amniotic fluid decreases. Okay. Enough. We shall let it be. Now we shall discuss this after lunch break. No problem. So we shall continue. Which of the following is not a derivative of mesonephric duct? One. Look at this diagram. In the initial stage of development, both the duct systems appear, develop in relation to the intermediate mesoidal. This is the undifferentiated state where we see the development of both mesonephric and paramesonephric ducts. These are the two mesonephric ducts. These are the two paramesonephric ducts. Both of them develop in the initial stage. Gonad is undifferentiated. We do not know whether it is going to become ovary or testis. The Y chromosome determines whether the gonad should become testis. There is a factor in the Y chromosome called Mullerian inhibiting factor. Mullerian inhibiting substance, also known as simply inhibin. This substance produced in the system due to the presence of Y chromosome results in regression of paramesonephric duct. Genetic sex male, gonads become testis and Mullerian ducts regress. The mesonephric ducts undergo further development in males. In the absence of Y chromosome, the gonad becomes ovary. There is no inhibin now, no Mullerian inhibiting substance. Therefore, the Mullerian duct undergo further development. Mesonephric duct regresses. You can see the two mesonephric ducts regress and the two Mullerian ducts undergo further development. So testis determining factor plus Mullerian inhibiting substance. This factor, Mullerian inhibiting substance, is actually produced by the Sertoli cells of testis. Sertoli cells of testis. Once the gonad is differentiated, if it is testis, then it will start secreting the inhibin, Mullerian inhibiting factor. That will result in regression of paramesonephric duct. Now look at this mesonephric duct. In males, it undergoes further development. It gives rise to parts of the male reproductive system. What are the things we shall see? epididymis, its continuation, vas deferens. This diverticulum, ureteric diverticulum, we have seen already, develops irrespective of the sex. Irrespective of the sex, the ureteric diverticulum develops. Then another diverticulum appears distal to it, and that diverticulum becomes the seminal vesicle. The duct of the seminal vesicle and the vas deferens unite to form the ejaculatory duct, which opens into the posterior wall of the prostatic urethra. So all duct system start right from the epididymis, vas deferens, seminal vesicle, ejaculatory duct. All those things develop from the mesonephric duct. The cranial end of the mesonephric duct is blind. This blind end becomes the appendix of epididymis. Appendix of epididymis. But appendix of testis is a remnant of paramesonephric duct, Mullerian duct. Appendix of epididymis is the cranial blind end. It appears as a small sessile body at the top of the head of epididymis. This is the genital ridge that becomes the testis. Medial swelling. Lateral swelling is the nephrogenic cord. We have seen that pronephros and mesonephros have disappeared. Only metanephros persists. So the right answer for our question is appendix of testis. The two Mullerian ducts fuse to form the uterus, and the remaining parts become the uterine tubes. Here there is a failure of fusion of right and left paramesonephric ducts. This can result in number 1. Look at this picture. Here we see the two Mullerian ducts. These two ducts fuse near their distal end, as you see here. The two distal parts of the two ducts fuse to form the uterus. So uterus is formed from the fusion of the two Mullerian ducts. The proximal parts, upper parts, remain separate. They do not fuse. They form the uterine tubes. So uterus and uterine tubes develop from paramesonephric duct. If the two paramesonephric ducts do not fuse, what will happen? Let us see. They remain separate. The distal part also remains separate. What will happen? Each distal part will become a uterus. So the person will have double uterus. This is what is called uterine diadelphys. Failure of fusion of the two Mullerian ducts will result in uterine diadelphys. Suppose the fusion occurs but only partial. Incomplete fusion occurs. What will happen? The two Mullerian ducts have come closer and partially fused. This will result in subseptate uterus. What is it? Externally you will see a single uterus. But if you examine the uterine cavity, it is divided into two compartments by a septum in the midline. This is what is called subseptate uterus. Externally a single uterus, but internally a septum will be separating the cavity into two compartments. This is uterus didelphes or uterine didelphes. Non-failure of fusion, non-fusion. Incomplete fusion will result in subseptate uterus. Okay. Descent of testis through the inguinal canal usually begins number 3, 7th month. You have to take 4 points to understand the descent of testis. Take 1 point at the deep inguinal ring, another point in the inguinal canal, third point at the superficial inguinal ring, fourth point in the scrotum. Deep inguinal ring, inguinal canal, superficial inguinal ring, scrotum. Now you fill or put this. 6th month, the testis lies at the deep inguinal ring. Around 6th month, passes through the inguinal canal. Around 7th month, reaches the superficial inguinal ring. Around 8th month, and reaches the scrotum finally by the end of 8th month or 9th month beginning. So put 6, 7, 8, 9 in that order, easy to remember. Take 4 points and put these numbers 6, 7, 8, 9. So it travels along the inguinal canal around 7th month of intrauterine life. It is there in the scrotum by the end of 8th month or the beginning of 9th month. So this is the right answer for our question. Failure of development of which of the following is a common cause of congenital diaphragmatic hernia? This hernia is also known as hernia of Bosdalek. These are the 4 structures contributing to the development of diaphragm. Failure of development of one of these results in the congenital diaphragmatic hernia, which is that one. 2. Then what? This picture diagram shows various structures contributing to the diaphragm. As we have seen already, as we have discussed, the septum transversum forms the central tendon of the diaphragm. The C-shaped tendon, central tendon. The peripheral part of the diaphragm is contributed by the mesoderm of the body wall. The posterior lateral aspects of the diaphragm, posterior lateral aspects, contributed by pleuroperitoneal membrane. Do you remember this membrane? This is the one that divides the pleural cavity from the peritoneal cavity. Periphery, mesoderm of the body wall. The gap between the two the area of the two pleuroperitoneal membrane is filled by the mesentery of esophagus, dorsal mesentery of esophagus. These are the structures contributing to the development of diaphragm. development of one of these is the reason for congenital diaphragmatic hernia, the hernia of Bose-Dallek. That is, you can see here. Sphenoid tendon has developed to a great extent, but you see there are two posterolateral defects. Abdominal contents herniate into the pleural cavity. This results in non-development of lungs. The child survives as long as it is inside the uterus because of placental circulation. But once it comes out, pulmonary circulation cannot be established. It cannot have independent circulation. Therefore, the child dies immediately after birth. Here you see the left lung has not developed at all. Hypoplastic left lung, right lung has developed to some extent. Need not be always. Both lungs fail to develop and the child dies immediately after birth. We can't do anything. These defects are due to failure of development of what? Pleuroperitoneal membrane, correct. Yes, metaphase, correct. It is at this stage the chromosomes are all arranged in the equatorial region of the cell. So chromosomes are isolated from the cell and are arranged. And are grouped like this. To which group in a karyotype the Y chromosome belongs? See, we have taken the WBC culture, mainly the lymphocyte culture. We have added the mitogen and nutritive medium, and the suspension, cell suspension is incubated at body temperature, that is 37 degrees Celsius for 2 days. Now the chromosomes start appearing in the lymphocytes, and when the chromosomes are all arranged in the equatorial region of the cell, we must arrest the cell division. If we allow the cell division to continue, the chromosomes will be separated and two daughter cells will be formed. Soon the chromosomes will disappear. In that area you will get a nucleus. So this we should not allow to occur. Therefore our idea is to see the chromosome. Our purpose is to see the chromosomes. So we must arrest the cell division at the stage of metaphase. There is a substance which inhibits the spindle formation. It is because of the formation of the spindles, the chromosomes are separated from the midline and two daughter cells are formed following the division of the cytoplasm. Which is the substance? Colchicine is added to inhibit the formation of spindles. Now the chromosomes are all seen within the cell. Then the cells are treated in hypotonic saline, potassium chloride. Why? The hypotonic saline will enter the cell and as a result the cell will become swollen. The cell membrane becomes quite thin. Now that suspension is taken in a pipette and with a slide keeping on the table or with your other hand, 1 or 2 drops of that fluid is dropped from that pipette. It should fall exactly onto the slide. It needs some experience. Otherwise it will not fall exactly onto the slide at a distance of 2 feet at least. Once the fluid falls on the slide, the drop falls on the slide, because the cell membrane has already made thin due to accumulation of hypotonic saline inside, the membrane ruptures and the chromosomes spread out onto the slide. Now we stain the chromosomes using Giemsa stain. This is the stain commonly chosen, protein stain for karyotyping. Used for karyotyping, Giemsa. And the chromosomes are identified based on 3 things. Based on their length, based on the position of the centromere in each chromosome, and based on the characteristic, unique banding pattern, dark and light bands produced by each chromosome. Look at this picture. Based on length, the first chromosome is the longest chromosome. And in the descending order of their length, the chromosomes are arranged. You can see becoming smaller and smaller, and finally the smallest chromosome is the Y chromosome in males, or the chromosome number 22 in others. This is one point. Number 2, the position of the centromere. We divide the chromosomes into 3 types: metacentric, submetacentric, and acrocentric. On what basis? Based on the position of the centromere. If you find the centromere exactly in the midline, exactly in the center, that chromosome is called metacentric chromosome. You can see that this is, that comes later. Okay. You can see the centromere here, very close to the center of the chromosome. If you find the centromere a little away from the midline, from the center or near the periphery, that chromosome is called submetacentric chromosome. If you find the centromere near the tip of the arm chromatid, that chromosome is known as acrocentric chromosome. In human beings, telocentric chromosomes are absent, which are found in plants and other animals. Telocentric means the centromere is found near the periphery, not exactly at the tip, but a little away from the tip. That is telocentric. So we have only 3 types of chromosomes. Then you can see the banding pattern when we use the stain. Even without staining also you can see the dark and light bands, which are unique. Each chromosome presents a unique banding pattern, light and dark bands, characteristic banding pattern. This is also taken into account in identifying these chromosomes. And the chromosomes are grouped like this: group A, group B, C, D, E, F, G. Chromosome Y belongs to the G group, and chromosome X belongs to the C group. X chromosome belongs to the C group, and Y chromosome belongs to the G group, last group, smallest. As I told you before, in these chromosomes 1, 2, 3, A group, the centromere is in the middle, center, metacentric. Here you see the centromere is a little away from the center, therefore submetacentric. In this group also, from 6 to 12, the centromere is a little away from the center, submetacentric. Here you see the centromere at the tip, so they are acrocentric. So locating the centromere itself requires some experience. After that, the banding pattern. Yes. Only through experience we can identify the individual chromosomes. Not all on a sudden, impossible. It requires a lot of training to identify the individual chromosomes. After identifying the normal chromosomes, we should be in a position to interpret any abnormality in those chromosomes like deletion, addition, translocation, etc. So it is a separate field. Medical genetics is a specialty, is a well established specialty in Western countries. Medical geneticist. And this is also a submetacentric chromosome, but very small. Submetacentric, acrocentric. You may see some small projections from the end. These are not the P arms, they are the satellite bodies. Some acrocentric chromosomes present two horns like this. They are called satellite bodies. They are exocentric chromosomes. They are not the P arm of the chromosome. P arm is the short arm, Q arm is the long arm of a chromosome. This is the Q arm. This is above the centromere, that is P arm. P, Q, alphabet. So, Y chromosome belongs to, yes. What is the formula? It is the total number of X chromosomes minus 1. N minus 1. N denotes number of X chromosomes. What is the chromosome constitution of a person with Klinefelter syndrome? 47. One extra chromosome is there. How? 47 XXY. Of the two X chromosomes, one X chromosome will be inactivated. Therefore, the inactivated chromosome appears as a darkly stained material known as bar body. In females, there are two X chromosomes. So one X chromosome is inactivated, and this process is known as lionization. It is because it was Mary Lyon who put forth this theory, lionization. Lyon hypothesis, L-Y-O-N, Lyon. Mary Lyon. So, normal females and Klinefelter male will have one bar body because one X chromosome will be inactivated. Normal males, they have only one X chromosome, they cannot be inactivated, they should remain, they should be active. And Turner's female. What is the chromosome sex of the Turner's female? 45X0. So there is only one X chromosome, already only one. So there cannot be spared. Therefore, normal male and Turner's female will not have any bar body. Bar body will be absent. What about a person having XXX chromosome constitution? Two bar bodies, yes, it is total number of chromosomes, X chromosomes minus 1. What are they called? Super females. But they are not actually super females. Most of them are mentally retarded. And the degree of mental retardation is directly proportional to the number of X chromosomes. More X chromosomes, more mental retardation. Only with extra X chromosomes, I am not talking about the normal X chromosomes. Extra chromosomes troublesome. So a woman with congenital dysmorphism is married to a normal man. What will be the fate of their children? To answer this question, we must know the mode of inheritance in congenital dysmorphism. Only then we will be in a position to find out the right answer for this question. What mode of inheritance is there in congenital deaf mutism? If you know that, we can answer this question. Autosomal dominant. Okay, then accordingly you find out the answer. If it is recessive, then accordingly you find out the answer for this question. Okay, I will tell you the mode of inheritance in congenital deaf mutism. It is an autosomal recessive disorder. See, if there is a defective gene, mutant gene in one autosome, and if the condition manifests phenotypically, that gene is a dominant gene. It is a dominant disorder. But a recessive gene cannot manifest if it is available in single dose. The other corresponding gene called allele, that also should have the same defect. When defective genes in both the autosomes, homologous pair of autosomes, are found in an individual, the disease will manifest. That is the feature of autosomal recessive disorder. One defective gene inherited from the father, other defective gene inherited from the mother. When two genes come in one person in the same chromosome, the disease will manifest. That is the feature of autosomal recessive disorder. On the other hand, if it is a dominant disorder, even if the person has one single defective gene in one chromosome, not in a pair of chromosomes, only one chromosome, the disease will manifest. That is the difference between dominant disorder and recessive disorder. Autosomal dominant, that is the defect is in the autosome. Sex-linked dominant, it is in the sex chromosome. That's all. That is the difference. But this is the thing. Congenital deaf mutism is an autosomal recessive disorder. And this woman is having the defect. That means she is already having two genes, two defective genes in the same chromosome. And she is married to a normal man who does not have any of the defective genes. No mutant genes in him. What will be the fate of their children? Father is normal. Mother is affected. She is having mutant alleles. You see, both the chromosomes are mutant, containing mutant genes. At the time of segregation, each child receives a defective gene from the mother and a normal gene from the father. So each child carries only a single copy of the defective gene. Will the condition manifest? But are they perfectly normal? No, apparently, phenotypically they are normal, but in them they have this defective gene in a single dose. So they are therefore called carriers. All of them are going to be carriers. Apparently normal, but they have a single copy of the defective gene. When two such apparently normal individuals marry, what will happen? 1:1/4. 1:1, 25% normal, 50% carriers and 25% affected. That will be the proportion. Sickle cell disease also is an autosomal recessive disorder. Congenital deaf mutism. So when apparently two normal individuals marry having sickle cell disease, that we commonly seen in some ethnic groups, because marriage between the same group is quite common and they are apparently normal, both male and females are normal, but when they marry, there is a chance, 25% chance of their children being affected, 50% remaining carriers and 25% normal. That is the pattern. So a deaf mute is often thrown into the company of other deaf mute, and a marriage between two such deaf mutes is not uncommon. When such a marriage occurs, what will be the fate of their children? A deaf mute marrying another deaf mute. All their children are going to be deaf mute because each parent is already having two genes. two defective genes. So all their children are going to have the two defective genes, and they are all going to be affected. So deaf mute marrying another deaf mute, right after their children, all are going to be affected. A carrier marrying carrier, 1:2:1, 25%, 50% into 25%. Normal, carrier, affected. That is the pattern. So all their children are going to be carriers, because each child receives only a single copy of the defective gene from only one parent. Normal ovary is perfectly correct. Normal ovary, female pseudohermaphroditism, therefore normal ovary, and external genitalia of the opposite sex. Not normal female external genitalia. The first system to develop in the embryo is central nervous system, but this is not the system to attain a functional state in the embryo first. Central nervous system develops because this is the system which has to coordinate all activities of the embryo. But the rapidly dividing neurons require lot of nutrients and oxygen, which cannot be met by this yolk sac alone in the beginning. So it needs an effective transporting system to carry the oxygen and nutrients to the rapidly dividing neurons. Therefore, the cardiovascular system becomes functional in the early period of development. Our heart develops early. Heart starts pumping blood early. When? The next question will tell. Not this one, after this. Now we know all four types of mesoderm. Heart, being a viscera, should develop from splanchnopleuric mesoderm. Splanchnopleuric mesoderm in the cardiogenic area of the embryonic disc. We shall see that area. This is the area, cardiogenic area, where the pericardium also is developing. In the beginning, okay, we shall take a section. Yes, you can see that reddish color in the splanchnopleuric mesoderm. This area, cardiogenic area, splanchnopleuric mesoderm. This is the area where we see the development of two endothelial heart tubes from which our heart develops. We shall discuss those things in detail. Two endothelial cores develop in that area due to proliferation of angioblastic cells of the splanchnopleuric mesoderm, as you see in this diagram. Two endothelial cores. Soon these cores are canalized to form two tubes. Canalized. Now the embryonic disc is folded upon itself in all directions. Lateral folding occurs. As a result of lateral folding of the disc, the two endothelial tubes come closer and fuse to form one heart tube. The fusion is complete in the cranial part, but incomplete near the caudal end. Fusion begins in the cranial end, progresses towards the caudal end. However, near the caudal end, the two tubes remain separate. So a single heart tube is formed by the fusion of the two endothelial tubes. Now, dilatations appear in the heart tube. Alternate areas are dilated. One dilatation, then constriction, one dilatation, one constriction. Like that, the tube presents alternate dilatations and constrictions. And we have given these names to the dilated part of the heart tube. Let us start from the cranial end. The cranial most dilatation is known as truncus arteriosus, TA. Just below that we see a small bulging called conus or bulbus cordis. Further below we see the primitive ventricle. Further below, atrium. At the caudal end, we see the sinus venosus. The two tubes which remain separate, they become the horns of the sinus venosus. Right horn, left horn, like that. Now we have ventricle and atria. Two ventricles and two atria. Do we have truncus arteriosus? No, it is only an embryological structure. Similarly, do we have sinus venosus? Do we have any horns? No. So they disappear later. Let us see how they disappear. If you examine each horn of the sinus venosus, you can see three veins joining the horn. Right common cardinal vein on the right side, left common cardinal vein on the left horn. Right umbilical vein and right vitelline vein. Left side, left umbilical vein, left vitelline vein and left common cardinal vein, like that. So each horn receives three veins. Right common cardinal vein from the upper part of the fetus, body of the embryo. Vitelline vein from the yolk sac. Umbilical vein from the placenta of mother. Umbilical vein through the placenta, common cardinal vein from the upper part of the body, vitelline vein from the yolk sac. Now let us see the fate of this embryonic heart tube. The truncus arteriosus, this name is given because it is the trunk of two major arteries arising from the heart. Which are the two arteries arising from the heart? Ascending aorta and pulmonary trunk. Ascending aorta from the left ventricle and pulmonary trunk from the right ventricle. So this is the trunk which gives rise to the two major arteries of the heart, truncus arteriosus. How it is partitioned into two with the development of a septum in between. Summary of development. Conus is not an adult structure. Therefore, it is absorbed into the ventricle. The conus bulbus cordis is absorbed into the ventricle and a common ventricle is there in the beginning. This common ventricle is divided into right ventricle and left ventricle with the development of intraventricular septum. So right and left ventricles develop from primitive ventricular chamber. The conus is absorbed into the ventricle. Similarly, the sinus venosus also is absorbed into the atrium along with its horns. And the common atrium is formed in the beginning. This common atrium is separated, is divided into right atrium and left atrium with the development of an interatrial septum. Right atrium, sinus venosus and the right horn all are absorbed to form one common chamber which is partitioned later into right atrium and left atrium. Then what happens to the left horn, you may ask? Left horn is greatly reduced in size and becomes the coronary sinus, the largest vein draining the heart. Coronary sinus is a vein, largest vein draining the heart. It opens into the right atrium. What is the fate of these veins which open into the horns of the sinus venosus? You shall see. Which are the veins opening into the right atrium in adult? Superior vena cava, inferior vena cava and coronary sinus. We have already seen how the coronary sinus develops. It is nothing but the left horn of sinus venosus. It regresses in size and becomes the coronary sinus. What about the superior vena cava and inferior vena cava? Let us see. Right common cardinal vein becomes the terminal part of SVC. Right umbilical vein disappears. In the beginning, umbilical cord has 4 blood vessels, 2 arteries and 2 veins. Later what happens? One vein disappears. Therefore the umbilical cord has 2 arteries and 1 vein. Which vein is left and which vein disappears? Right umbilical vein disappears and left umbilical vein is left. Left is left. In that way, we have 2 umbilical arteries and 1 umbilical vein in the latter stage of pregnancy in the umbilical cord. The right vitelline vein carrying blood from the lower part of the body. What is it? It becomes the terminal part of IVC. So 3 veins open into the right atrium: SVC, IVC and coronary sinus. SVC, right common cardinal vein; IVC, vitelline vein; coronary sinus from the left horn. All are absorbed into the right atrium. That is how these 3 veins open into the right atrium in adult. So this is the summary of development of heart. You can see the bending of the heart tube. So it has to retain the adult shape. The adult shape should be attained. For that, this is what happens. You see, there is a bending here. T denotes truncus arteriosus, B for bulbus cordis, V for ventricle, A for atrium, SV for sinus venosus. Now a sulcus appears between the bulbus cordis and ventricle. This is called bulboventricular sulcus. Leave it. Now you see, this sulcus gradually disappears and bulbus cordis and ventricle become one chamber. Now, with the disappearance of this sulcus. Now the atrium grows backwards behind the truncus arteriosus and is partitioned into right atrium and left atrium. Now this is the common ventricle following absorption of the bulbus cordis with the disappearance of the sulcus. Now there is a common ventricle. This common ventricle is partitioned into right ventricle and left ventricle with the development of interventricular septum. Similarly, as I told you before, the truncus arteriosus also is divided into 2 parts like this. One part becomes the ascending aorta, other part becomes the pulmonary trunk. Right ventricle, left ventricle with the development of interventricular septum. Right atrium, left atrium with the development of interatrial septum which lies behind. So atria lie above and behind. Ventricles lie below in front. As you see, this is the position of our adult heart. 2 atria lie above and behind. Not only above but behind. 2 ventricles lie in front and below. Okay. So truncus arteriosus means ascending aorta and pulmonary trunk. Common ventricle means the bulbus cordis plus primitive ventricle. Common atrium means primitive atrium plus sinus venosus and the horns. So heart develops from splaganopleuric mesoderm in the cardiogenic area of embryonic disc. As I told you before, heart starts pumping blood from the early period of life. When does it start beating? fourth week. By the beginning of the fourth week. Perfectly correct. 22nd day or 23rd day of pregnancy. It starts pumping blood right from 22nd day or 23rd day of pregnancy. Goes on beating, even now beating. One day it will stop. With that everything will come to an end. We see this fossa in the right atrium, in the right atrial septum on the right side. It represents one of these embryonic structures. Which is that one is our question. Septum primum, septum secundum. Look at this. These diagrams are there to show you the development of interatrial septum. Interatrial septum develops from two septa, primarily from two septa, septum primum and septum secundum. This is the septum primum and this septum is the septum secundum. The upper margin of the septum primum is quite thin and movable like a flat valve. The lower margin of the septum secundum is quite thick and rigid, it does not move. The oblique passage between these two septa is the foramen ovale. So foramen ovale is an oblique passage between these two septa during development. Septum primum because it is the first septum to develop. The other septum develops later, that is why septum secundum next to the primum. Now during intrauterine life, the pressure, more blood reaches the right atrium but only small amount of blood reaches the left atrium because pulmonary circulation is not established. So what happens? Pressure in the right atrium is greater than the pressure in the left atrium. Therefore, the blood pushes the septum primum away from the septum secundum and thereby this foramen ovale is very much enlarged. Blood can easily flow from the right atrium to the left atrium. But if the blood tends to flow in the opposite direction, the septum primum is pushed towards the septum secundum and it closes the foramen ovale. So, blood is permitted to flow only in one direction, from the right atrium to the left atrium, not the other way around. Any tendency of the blood to flow in the opposite direction will result in closure of the foramen ovale. The two septa will move towards each other and the foramen ovale will be closed. But what happens immediately after birth? Pulmonary circulation is established, more blood reaches the left atrium. Now this more blood, the pressure in the left atrium becomes greater than the pressure in the right atrium. Now the septum primum is pushed by the blood and it is pushed towards the septum secundum and the two septa come closer, lie in opposition and finally fuse. Now the foramen ovale is closed forever. That is how the foramen ovale is closed. In that area of closure, we see two structures, fossa ovalis and limbus fossa ovalis, are the two parts that we see in the interatrial septum. I will show you that. This is that. We have removed the wall of the atria and this is the interatrial septum where you can see a shallow fossa called fossa ovalis. And it has a thick margin here. That thick margin is the limbus fossa ovalis or annulus ovalis. These two parts represent the two septa, septum primum and septum secundum of the embryonic heart. Fossa ovalis, if you apply logic, represents which of these? Septum primum, correct. Because it is thin, that is why it becomes a fossa. But septum secundum is quite thick. You see the lower margin, free margin of the septum secundum is quite thick. Therefore, it can form only a thick margin. Therefore, septum secundum, fossa ovalis, limbus fossa ovalis becomes the septum secundum. Septum secundum becomes the limbus fossa ovalis. Now you can see the real picture. We have cut and reflected the anterior wall of the right atrium. This is the area where we see the fossa ovalis and limbus fossa ovalis. Also you can appreciate the three openings in the right atrium, SVC, IVC and coronary sinus. The anterior wall of the right atrium is quite rough because of several muscular ridges. These muscular ridges are called the musculi pectinati. Now we will label, this is the fossa ovalis and this margin is the annulus ovalis or limbus fossa ovalis. This is the opening of IVC, opening of coronary sinus and opening of SVC. This is the right AV orifice, atrioventricular orifice, between the right atrium and right ventricle, tricuspid orifice. This is what happens after birth. Foramen ovale is closed, septum primum becomes the fossa ovalis and septum secundum becomes the limbus fossa ovalis. You are seeing a large defect in the atrial septum. It's a case of ASD. There are different types of ASD, atrial septal defect. But this one is quite common. It is more common in females, ASD, but VSD is more common in males. The most common form of cardiac anomaly is VSD, ventricular septal defect, more commonly seen in males. Here you are seeing a defect. This is because of shortening of the septum secundum. The two septa do not overlap. Only when they overlap, then the whole thing can be closed. But if the septum secundum is quite short, what will happen? After birth, a large gap will be present in the interatrial septum. This type of ASD is quite common and is called secundum ASD. Most common form of ASD. Secundum ASD is there, primum ASD is there, sinus venosus ASD is there. There are different types, but this is the one which is common, so I am not going into other details. So fossa ovalis of the adult heart represents the septum primum. Even before birth, sometimes the foramen ovale may close. If it closes, which of these is a possibility? You must apply logic and find out the answer for this. Foramen ovale is closed. Now the blood cannot flow into the left atrium from the right atrium. So the entire blood in the right atrium has to flow into the right ventricle. Right ventricle receives more blood. So it has to pump the blood into the pulmonary trunk. It has to do more work to empty the right ventricle. So what will happen to the right ventricle? That is what is happening. So right ventricular hypertrophy, it will result in right ventricular hypertrophy because it receives more blood now. It has to undergo hypertrophy to empty the blood. The interventricular septum has two parts, a lower muscular part and an upper membranous part. The interventricular septum develops from these three things that we will see in the diagram. AV cushion, also known as atrioventricular cushion, endocardial cushion, bulbar septum, primitive ventricular chamber. These are the three things contributing to the development of interventricular septum. The membranous part of the septum develops from That is the question. Number 1. Want to go with number 1? Look at this. This is the lateral view, side view. Atrium is here, ventricle is here, common ventricle. Ventricle means common ventricle following the absorption of bulbous cordis. Bulbous cordis will be seen somewhere here. The ventricular septum develops from 3 sources. From the primitive ventricle, a septum extends upwards. This septum comes from below, therefore called septum inferior. It extends from the floor of the common ventricle, ventricular chamber. It grows upwards. Second source, we see in the bulbous cordis 2 swellings appearing, right and left swellings in the bulbous cordis. And soon these 2 swellings approach each other and fuse to form a septum, which you can see here. This septum is called bulbar septum, also called proximal bulbar septum, to differentiate it from other septum. This septum grows downwards towards the septum inferior. We will see that. See, it grows downwards, septum inferior grows upwards. They grow towards each other, but however they do not meet. They remain separate. There is a gap here now. This is the septum inferior, this is the proximal bulbar septum, and you see there is a gap here. This gap is filled by proliferation of tissue from the atrioventricular cushion. This is a cushion, atrioventricular tissue between the atrium and the ventricle. That is why it is called atrioventricular cushion or AV cushion. It lies between the atrium and the ventricle. Fused AV cushion, cells proliferate and grow towards, extend towards the gap between these 2. You are seeing that in this picture. Extension of tissue from AV cushion to fill the gap. And finally these 3 things fuse to form the interventricular septum. So ventricular septum develops from 3 sources: septum inferior, bulbar septum, and extension of tissue from AV cushion. The membranous part of the septum develops from AV cushion, and muscular part of the septum develops from septum inferior and also bulbar septum. You can see that. Proliferation of tissue from AV cushion. Now you are seeing a small gap in the membranous part of the interventricular septum. What is it? A case of membranous VSD. This is due to, now you know the reason, embryological reason behind. Failure of extension of tissue from AV cushion is the reason for membranous VSD. There are 2 types of VSD, membranous VSD and muscular VSD. Muscular VSD is due to incomplete development of septum inferior. You can see that, okay, this is another view of the same thing. Now you see a gap in the muscular part of the septum. This is muscular VSD, often due to defective development of septum inferior. In some individuals, both membranous and muscular VSD will exist together. Can you see that? This is membranous muscular. Small openings can be seen in the muscular part of the septum. What is that appearance called? Swiss cheese appearance. Swiss cheese appearance. This one. Swiss cheese appearance. Small openings will be found in the muscular part. Surgical intervention is not required. Usually these small gaps will fuse after 1 year of life. No surgical intervention. So VSD is the common anomaly. Muscular VSD, membranous VSD are both. Sometimes the septum fails to develop. That also we must expect. Failure of development of intraventricular septum. This results in a 3-chambered heart, 1 common ventricle and 2 atria. What is the term used for this? Cor biloculare. Cor triloculare. Triloculare means 3-chambered heart. Cor means body, 3-chambered body. How 3-chambered? Biatriatum with 2 atria. There is thorough mixing of both oxygenated and venous blood in the ventricle in this case. It is the mixer blood that travels everywhere. Look at the common ventricle. You are seeing all 4 orifices in 1 chamber. This is the pulmonary trunk. You can see the pulmonary orifice. This is the ascending aorta. You can see the aortic orifice. These 2 semilunar orifices. And this is the tricuspid orifice. And this is the bicuspid orifice. So all 4 orifices in 1 chamber. Thorough mixing of blood. So membranous part of the intraventricular septum develops from, now you know the answer. Yes, heavy question. Defective or failure of development of AV cushion can result in. You see, the AV cushion is not only responsible for the development of membranous part, but also it contributes to the lowermost part of the interatrial septum. Look at this picture. This is the AV cushion. It occupies the center of the heart, contributing to both the atrial septum and ventricular septum. If this part does not develop, what will happen? It will result in both ASD and VSD. You can see that ASD, VSD. So defective development of AV cushion. AV cushion is actually is due to migration of cells from the neural crust. Failure of development of cells from the neural crust can result in AV cushion defects. That is why, in trisomy 21, Down syndrome, Down babies, both craniofacial anomalies and anomalies of the heart coexist. Why? Because craniofacial anomaly, neural crust contributes to several skeletal and muscular elements of head and neck. It also contributes to the development of heart. That is why in Down syndrome, there is a defective development and defective formation and migration of neural crust cells. That results in both craniofacial anomalies and anomalies of heart, as we see in Down babies. Atrioventricular septal defect. This is also called cor biloculare because because of the defects, 2 atria communicate and 2 ventricles communicate. Net effect, 1 chamber above, 1 chamber below. So net effect, 2-chambered heart. That is why cor biloculare. So ASD and VSD. TGA or TGV, transposition of great vessels, is another congenital anomaly. In this case, ascending aorta originates from right ventricle, pulmonary trunk originates from left ventricle, carrying the oxygenated blood back to the lung, and ascending aorta carries deoxygenated blood, reduced blood to all parts of the body. Their position is just reversed, transposition of great arteries. We have already studied the structure from which these two arteries develop. What is it? Truncus arteriosus. So, it is due to one of these, transposition of great arteries. Which one you want to choose? Number 2, bulbar septum to follow a spiral course. Okay, we shall see. We have seen this diagram before. This is as we have seen already, a septum developing in the bulbus cordis. We also see two swellings appear in the truncus arteriosus. This is TA, this is bulbus cordis, ventricle, atrium, and behind that sinus venosus will be there. So just we have seen in the bulbus cordis, these two swellings approach each other and fuse to form another septum. This septum is called distal bulbar septum. And this distal bulbar septum fuses with the proximal bulbar septum. And this common septum is called aortico-pulmonary septum, cono-truncal septum, also known as spiral septum. Aortico-pulmonary. Why? Because it forms the aorta, it helps in the formation of aorta and pulmonary trunk. Spiral septum, this septum undergoes a spiral course, just like a spiral staircase. That is why it is called spiral septum. It is also known as conotruncal septum because it develops in the conus and also truncus arteriosus. That is why conotruncal. Cono, this is cono, this is truncal. Proximal cono, distal truncal. Conotruncal septum. Let us see how this septum follows a spiral course. Here you are seeing a tube inside which a septum, which separates the tube into two equal halves. Suppose this septum undergoes a spiral course. Let us see what happens. You trace this arrow, blue arrow. It is now seen on the right side of the red arrow. If you trace it up, it comes to lie on the left side of the red arrow. This is what normally happens in the development of ascending aorta and pulmonary trunk. Assume that this is the aorta. Aorta lies to the left of the pulmonary trunk at its beginning. When you trace it up, it comes to lie to the right of the pulmonary trunk. Similarly, the pulmonary trunk also runs like this. This is the adult situation. Pulmonary trunk to the left, but if you trace it up, it comes to lie to the left. It is now right, here it is to the left of the aorta. This is the normal thing. It is because of the spiraling of the septum. That is how pulmonary trunk begins from the right ventricle, ascending aorta begins from the left ventricle. Now let us see a situation where the septum fails to follow a spiral course. What will happen? Let us see. This is the normal one. Pulmonary trunk begins from the right ventricle, aorta begins from the left ventricle due to that spiraling of the septum. Suppose it fails to pursue a spiral course, instead it descends straight. What will happen? This is what happens. Now the aorta originates from the right ventricle, pulmonary trunk originates from the left ventricle. Aorta carrying the deoxygenated blood to all parts of the body, pulmonary trunk carrying the oxygenated blood back to the lung. Can a person survive? No. But person survive because the pulmonary septum, we have already seen, bulbar septum contributes to the intraventricular septum. Here there is, this septum has not undergone a spiral course. Therefore, it is not aligned with the intraventricular septum. As a result, a VSD always will be present. This is actually a boon. Because of VSD, mixed blood only will travel everywhere. Then survival is possible because of VSD. If VSD is not there, survival is not possible. Death will occur immediately. But it is the mixed blood that travels everywhere due to a VSD. So a TGA will be always associated with a VSD. So failure of the aorticopulmonary septum to follow a spiral course is the reason for TGA or TGV. TGA, transposition of great arteries. TGV means transposition of great vessels. Both are one and the same. Transposition of great arteries. This is what is called TGA. The mechanism is this: failure of spiral course. We have seen this septum, bulbar septum, dividing the truncus arteriosus into two equal halves. That is why the caliber of both the ascending aorta and pulmonary trunk equal. Suppose the septum deviates to one side, one artery becomes narrow, other artery becomes quite large. So what will happen if the truncus arteriosus is divided unequally? What will happen? Number one, it deviates to one side. Unequal division of truncus arteriosus. Now the pulmonary, this has deviated towards the pulmonary trunk side. Therefore, the pulmonary artery is very much narrowed, but ascending aorta is quite large. Valvular stenosis of the small vessel, say pulmonary stenosis. The aorta is quite large, it cannot be contained within the left ventricle, therefore it overlaps the right ventricle. Overriding of large vessel. Since the septum is not exactly in the midline, it has deviated to one side, it results in what? A VSD, ventricular septal defect. Now the right ventricle receives blood not only from the right atrium but also from the left ventricle through this defect. Therefore it has to do more work to empty the blood through the narrowed pulmonary artery. So it has to do enormous work. It receives blood from two sources now, not only from right atrium but also from left ventricle. But that enormous large quantity of blood must be emptied through a narrow vessel. See, pulmonary stenosis. So it has to do enormous work. It undergoes hypertrophy. So pulmonary stenosis, overriding of aorta, ventricular septal defect and right ventricular hypertrophy. These four are the features of tetralogy of Fallot. So it will result in tetralogy of Fallot. Simply because of a slight deviation from the normal path. That's all. Here you see pulmonary stenosis, overriding of aorta, thickness. You see the volume. Which is the ventricle that has the thickest volume of myocardium? Left ventricle. Now you see here the right ventricle is very much thick. right ventricular hypertrophy, VSD cannot be viewed in this case. VSD. These four things will occur. A slight deviation from the normal path. So, normal path is very, very essential in everything. Tetralogy of Fallot. If this is associated with ASD, then it is called pentalogy of Fallot. Sometimes the spiral septum fails to develop. What will happen? That also you must expect. It fails to develop. What will happen? Very simple. Yes. Look at this. The truncus arteriosus is not now separated into two arteries due to failure of development of the septum. So we see a large vessel, a single vessel, which opens both into the right ventricle and also into the left ventricle. This is what is called persistent truncus arteriosus, PTA. PTA, Parent Teachers Association. The arch of aorta and pulmonary arteries are normal here. Why? Because they develop from aortic arches, from the pharyngeal aortic arches. That is why they are normal. Again, the person can survive because of, look at the picture and tell, VSD. That is a bone. Number 2, if you know the fetal circulation, we can answer this question without any difficulty. Prenatal circulation, you see, this is the arch of aorta and this is the pulmonary trunk. These two are connected through a temporary channel during fetal life. What is that called? Ductus arteriosus. Due to this, the blood from the pulmonary trunk, what blood? Oxygenated or deoxygenated? Deoxygenated blood. It cannot reach the lung because lungs have not developed and are not functioning. Therefore, that blood returns and that blood should be somehow emptied from the system. Therefore, what happens? This connection helps to empty the blood into the arch of aorta. So, arch of aorta receives the deoxygenated blood, where after giving of these three arteries, 1, 2, 3, these are the three arteries of the arch of aorta. Which are they? Brachiocephalic trunk, left common carotid, left subclavian. So, ductus arteriosus opens into the arch of aorta distal to the origin of these three arteries. So, mixed blood is there in the aorta distal to these three arteries. And we know these three arteries supply the head and neck and our upper limb, brachiocephalic, common carotid, subclavian. They supply the upper part of our body. So, upper part of our body gets better oxygenated blood than the lower part of our body. Why? Because our brain is highly sensitive to hypoxia and hypoglycemia. It requires a lot of oxygen and glucose. Though the adult brain is only 2% of our body weight, but it consumes 20% of oxygen intake by the body. 20% of oxygen intake by the body. 20% is utilized by the, consumed by the brain itself. 80% to all other things. But it is only of 2% weight. So So it is in great demand for oxygen and nutrients. And a person can maintain consciousness only when there is a continuous cerebral blood flow. If the blood flow to the cerebrum, if the blood flow to the brain stops, the person becomes unconscious within 10 seconds. And irreversible brain damage occurs within 10 minutes if blood flow is not restored. Look at this structure. This is the blood vessel carrying oxygenated blood from the placenta of the mother. Which blood vessel? Umbilical vein, correct. Here umbilical vein carries the oxygen and nutrient rich blood from the placenta of the mother. Which umbilical vein? Left umbilical vein. This blood is poured into the inferior vena cava. You know, inferior vena cava carries deoxygenated blood from the lower part of the body. But in the fetal circulation, it has an entirely different work. It receives the oxygenated blood from the placenta of the mother through the left umbilical vein. This blood enters the right atrium. What is this temporary channel that connects the left umbilical vein with the inferior vena cava? Ductus venosum. Ductus venosus. And this blood is poured into the inferior vena cava, and inferior vena cava empties the blood into the right atrium. And this blood being oxygenated is not flowing into the right ventricle. It is pumped into it flows into the left atrium through foramen ovale. That is the purpose for which such an opening is made in the interatrial septum during intrauterine life. This oxygenated blood should somehow reach the left side of the heart to be pumped into the all parts of the body through the aorta. That is how the oxygenated blood reaches the left atrium. In adult, oxygenated blood reaches the left atrium through the 4 pulmonary veins from the lungs. Such a thing is not possible now. Therefore, the oxygenated blood reaches the left atrium through the foramen ovale from the inferior vena cava, whereas superior vena cava blood enters the right ventricle. It is deoxygenated. So, right ventricle receives mainly the blood from the superior vena cava. Right ventricle. But left atrium receives the oxygenated blood from the right atrium through the inferior vena cava. So, this deoxygenated blood in the right ventricle enters the pulmonary trunk, and the pulmonary trunk empties the blood into the arch of aorta. That is how these things occur. After birth, what happens? These are the 2 umbilical arteries which carry the deoxygenated blood back to the placenta. They carry the deoxygenated blood from the fetus to the mother. Umbilical arteries. These 4 structures regress after birth. Ductus arteriosus, ductus venosus, left umbilical vein and umbilical arteries degenerate. Let us see. The ductus arteriosus becomes ligamentum arteriosum. Ductus venosum becomes ligamentum venosum. And left umbilical vein becomes the ligamentum teres of liver. And the 2 umbilical arteries become the medial umbilical force or medial umbilical ligaments. These are the 4 vestigial structures of circulatory system, fetal circulatory system. With the establishment of pulmonary circulation after birth, all 4 regress. Now, in some individuals, this connection between the pulmonary trunk, especially the left pulmonary artery and the arch of aorta persists. It is patterned now, even after birth. What is this condition called? PDA, patent ductus arteriosus. It gives a continuous machinery murmur on auscultation. Surgically it can be corrected. Patent ductus arteriosus. So which blood vessel can carry blood with highest oxygen content? Need not be common carotid, brachiocephalic trunk, subclavian artery. All arteries of the upper part of our body carry better oxygenated blood because the arch of aorta receives the deoxygenated blood from the pulmonary trunk through the ductus arteriosus distal to the origin of these arteries. Pharyngeal arches. See, in the beginning, head develops and trunk develops. There is no neck. In the region of the future neck, small swellings appear. 6 swellings on either side of the future neck appear. 6 swellings this side, 6 swellings this side. Then what happens? They grow towards each other anteriorly and fuse to form 6 arches. Arch is complete anteriorly, deficient posteriorly. And these are the pharyngeal arches or branchial arches. 6 swellings this side, 6 swellings this side. They grow towards each other and fuse to form 6 arches. Arch is complete ventrally, deficient posteriorly, dorsally. Look at this picture. These are the swellings which appear in the future neck. This is the head swelling, this is the trunk swelling, and in the region of the future neck we see these pharyngeal arches, 6 in number. However, the 5th arch disappears soon after development. So for all, practically we have only 5 arches. First arch, second arch, third arch, fourth arch, fifth is not there, 6th arch. Like that we have only 5 arches. If you cut and examine each arch, you can see 4 types of tissue in it. What are the 4 types of tissue? Muscles, bone, blood vessels and nerves. You need not strain your memory to remember this because these are the 4 things our body is made up of. Muscle, bone, blood vessels, nerves. So our exercise is to know the derivatives of all these things because questions have been asked repeatedly in various examinations. In our first question we shall discuss the skeletal elements, skeletons, cartilaginous elements developing from these arches. You can see the arches viewed from behind. 4 components are there, 1, 2, 3, 4. Arch is complete, ventrally, deficient, posterior. That is why it is called arch. We are viewing this arch from behind. Yes, this is again to show you the same thing. Okay. What are the derivatives, cartilaginous derivatives of these arches? Let us take the first arch now. First arch cartilage. Which are the bones developing from first arch? The most important bone is mandible. And this cartilage that we see inside the mandible is the Meckel's cartilage. Then a ligament extending from the spine of the sphenoid to the mandible. What is it called? From the sphenoid to the mandible? Sphenomandibular ligament. And 2 ear ossicles. Malleus. Ready? Ellarukku? T. Incus. These are the 4 structures derived from first arch. Mandible, one ligament, two small ear vesicles. Let us see the derivatives of the second arch cartilage. Which ear vesicle remains now? Okay, stapes. There is a long slender process extending from the base of the skull. What is it called? Styloid process. From which a ligament extends to the hyoid bone. Stylohyoid ligament. Which horn of the hyoid bone is this? Small horn. And what about this part called body? Which part of the body now develops from this? Superior part of body and small horn. Small horn and superior part of body of the hyoid. Did you notice any peculiarity about the structures derived from the second arch? They all begin with the letter S. Stapes, styloid process, stylohyoid ligament, small horn and superior part of body. Don't say lesser horn and upper part of body. S will not be there in that situation. Though you are right. To bring that S, you must say small horn and superior part of body of hyoid. If you could remember the derivatives of these two arches, you need not strain your memory to know the derivatives of the rest of the arches. Those things will come automatically. What is remaining in the hyoid bone? Greater horn and lower part of the body. They develop from third arch. So all you have to do is to remember the derivatives of the two arches only. Now all cartilages of the larynx develop from rest of the arches. Simple. Fourth and sixth arch. Cartilages of the larynx. All cartilages of the larynx develop from fourth and sixth arch. So a single diagram illustrates all the derivatives of the pharyngeal arch cartilages. Now which one you want to choose? Greater horn of hyoid bone. Instead, smaller horn of hyoid bone. To bring that S. Lesser horn. This we have already discussed. Facial nerve. Okay. Men are inside the cage. Animals are outside. Tea is ready. We shall have a short tea break and assemble after 5 minutes. So we shall continue. The branchial cyst is due to persistence of. Do you know the answer for this question? Number 4. 1. 1 or 4? 1. Majority. 4 also equal. Look at the pharyngeal arches in section. First arch, second arch, third arch, fourth arch and sixth arch. These are the 6 arches on the right and left side. If these arches persist, the side of our neck will not be smooth. It will have 5 swellings this side, 5 swellings this side. Just like our extraterrestrial one, ET1 movie. That extraterrestrial being, has 5 swellings this side and 5 swellings this side. Only after knowing embryology only they might have designed that creature. But our neck is quite small. How this happens? You see, a tissue flap extends from the second pharyngeal arch. This goes down, extends down, and fuses with the side of the neck below these arches, giving the side of the neck a smooth appearance. This tissue flap from the second arch grows down and fuses below the arches with the side of the neck. When it goes down, you can see a gap between the tissue flap and the side of the arches. This gap is known as cervical sinus. Once the tissue flap fuses with the side of the neck, this gap completely disappears. No trace of it will be found later. But in some individuals, rarely, this gap may persist. If it persists, it can give rise to small cyst along the anterior border of the sternocleidomastoid. It can result in branchial cyst or branchial fistula or branchial sinus. You can see one photograph. You can see usually the branchial cyst, sinus and fistulae, they all lie along the anterior border of the sternocleidomastoid. All because of persistence of cervical sinus. Cervical sinus is the right answer for this question. Second pharyngeal arch is there, but that is not the reason for the development of this. It is because of the persistence of the gap between the tissue flap and the arches. Now we are going to see the development of eustachian tube, middle ear, palatine tonsil, parathyroid, thymus, all those things. The gaps between the arches, internal gap between the arches, is known as pharyngeal pouch. Similarly, gaps are there externally between the arches. They are called ectodermal cleft. Which pharyngeal pouch is associated with the development of inferior parathyroid is our question. Look at this. Between 5 arches, we get 4 gaps. This is the gap between the first and second arch, first pouch. This is the gap between the second and third arch, then third and fourth arch, and fourth and sixth arch. So 4 pouches are present between 5 arches internally. And these are the ectodermal cleft. First ectodermal cleft, second ectodermal, third, fourth, like that. External gaps are called ectodermal cleft. Internal gaps are called pharyngeal pouches. So between 5 arches we have 4 internal gaps and 4 external gaps. These pouches are associated with the development of several important structures in our body. You see the first arch, first pouch between the first and second arch. This extension found between the first or in the first pouch is known as tubotympanic recess. Tubotympanic recess. The name clearly suggests the structures which develop from this extension. Tube, that is eustachian tube. Tympanic, that is tympanic cavity, that is middle ear. Inner surface of the eardrum, master antrum and air cells, everything in that part. So tubotympanic recess gives rise to, that is why the eustachian tube opens into the nasopharynx. It is the communication between the nasopharynx and middle ear. Middle ear itself develops from the first pharyngeal pouch. Master antrum, air cells, inner surface of the eardrum. This one, this part, this is the eardrum, tympanic membrane, inner surface. The outer surface of the tympanic membrane and the external acoustic meatus and outer surface of the tympanic membrane develop from first ectodermal cleft. The inner connective tissue element of the tympanic membrane develops from mesoderm. So now you can understand one thing. Tympanic membrane is one structure in our body getting contribution from all 3 germ layers. Inner surface from the endoderm, outer surface from the ectoderm, and the middle or connective tissue part from the mesoderm. The second pouch gives rise to the palatine tonsil. Third pouch gives rise to 2 structures, the inferior parathyroid and thymus. These 2 structures develop from third pouch. Which parathyroid remains now? Yes. Superior parathyroid arises from the 4th pouch. From the picture, inferior parathyroid lies above the superior parathyroid. Is it right? No. So inferior must be inferior. Now it is superior. It is placed up above. So what happens? The inferior parathyroid is dragged by the thymus, because we know thymus is a structure of the thorax. It has to migrate into the thorax. When it migrates into the thorax, it drags the inferior parathyroid also along with it. Once the inferior parathyroid comes well below the superior parathyroid, that means somewhere here below the superior parathyroid, it leaves the inferior parathyroid there and goes alone into the thorax. That is why the position of the inferior parathyroid in adult often varies, because it always develops along with the thymus. Thymus migrates, this also migrates, therefore its position varies. But the position of the superior parathyroid is relatively constant on the posterior surface of the thyroid gland. This inferior parotid may be found in the jugular node in some cases, or it may be found in the thorax also. Possible. Because it moves along with the thyroid, the thymus. That's what is shown in this picture. We will skip this one. Okay. So inferior parathyroid now develops from third pouch. Correct. Now comes the vascular component of the pharyngeal arches. They are called aortic arches simply. 6 arches are there, so 6 arteries appear. 1 artery in each arch. The 5th arch itself disappears, therefore there is no artery for the 5th arch also. But unlike the main arch, more than 1 arch artery disappears during later period of development. In this question, the following arteries disappear except 3. Look at this picture. 6 arteries, 1st, 2nd, 3rd, 4th, 5th, 6th. 6 arch arteries appear in the beginning. Later, the 1st and 2nd arch arteries disappear, 5th or even otherwise disappears. The main arch itself disappears, so that you can easily remember always. But in addition, the 1st and 2nd also disappear. So we have only 3 arch arteries. These 3 arch arteries give rise to all arteries of the head and neck and thorax. All arteries of the head and neck and thorax are derived from these 3 arches only. Our exercise is to know the various arteries developing from these 3. 3rd, 4th and 6th. Here in this, all disappear in this question except 3rd, correct. 3rd one persists. 4th also persists and 6th also persists. The 6th aortic arches give rise to any idea? Pulmonary trunk. Pulmonary arteries. Not trunk anyway. Pulmonary trunk is from truncus arteriosus. Only 3 arteries are shown in this picture. 3rd arch, 4th arch, 6th arch. The 3rd arch gives rise to the common carotid artery. Left side, left common, right side, right common carotid artery. The 4th arch gives rise to the arch of aorta on the left side, right subclavian artery on the right side. 6th arch, from the color itself you can easily understand. 6th arch gives rise to the pulmonary artery, left pulmonary artery left side, right pulmonary artery right side. These 2 arteries establish communication with the arteries developing in the lung bud. This is the site of communication. But you know, from the picture you can understand one thing. The 6th arch artery communicates with the 4th arch artery through this connection. This connection disappears on the right side, but on the left side it persists. What is it called? This is the ductus arteriosus. It is the link between the 4th arch artery and the 6th arch artery on the left side. Such a communication disappears on the right side. So 3rd arch means common carotid artery should flash in your mind. 6th arch means pulmonary artery should flash in your mind. But for the 4th arch, you should remember 2 things. Left side, arch of aorta, right side, right subclavian artery. So right subclavian artery is an artery of the 4th pharyngeal arch. Remember. Left subclavian artery arises from the intersegmental artery, 7th cervical intersegmental artery. It is not from the pharyngeal arch, local artery. Now the answer is pulmonary. Suppose you want to choose number 1. What modification will you make? Okay. That is the question. Suppose you want to choose number 2, arch of aorta, what modification will you make? Fourth arch artery on the left side. It should be like that. The same arch on the right side gives rise to the right subclavian. Okay. Development of nervous system, brain. One diagram is enough. Pons and the cerebellum develop from no idea. We have already seen the development of neural tube. In the beginning, it is not a closed tube. It is open cranially and also caudally. They are called cranial neuropore and caudal neuropore. If these neuropores remain open, ventricular system cannot develop. Cerebrospinal fluid cannot be contained. So if the ventricular system does not develop, brain cannot develop. If brain does not develop, head cannot develop. That will result in anencephaly. So these neuropores must be closed. The cranial neuropore closes first when on 25th day of pregnancy. Caudal neuropore closes 2 days later, 27th day. The closure of the two pores, the closure of the cranial neuropore actually begins in the neck region, cervical region, and progresses towards the head side. Failure of closure of this neural tube will result in several neural tube defects. Spina bifida, meningocele, meningomyelocele, and all those things. Folic acid deficiency is one of the main reasons for development of neural tube defects. Therefore, it must be supplemented with the before pregnancy or after pregnancy. Before pregnancy, then only it is useful. Now the two neuropores are closed. Ventricular system has developed. As a result, the neural tube dilates into 3 vesicles: forebrain vesicle, midbrain vesicle, and hindbrain vesicle. Now it is a closed tube. 3 dilatations appear in the neural tube: forebrain, midbrain, hindbrain. The other terms used for these 3: forebrain means prosencephalon, midbrain means mesencephalon, hindbrain means rhombencephalon. Soon, 2 lateral swellings appear in the prosencephalon. Look at this. 2 lateral swellings appear in the prosencephalon. Now the prosencephalon has 3 swellings: right and left swellings and a midline swelling which was there before. Now 2 terms are used for this. The lateral swelling is called telencephalon, and the midline swelling which was there in the beginning is now known as diencephalon. Mesencephalon does not undergo any change, but rhombencephalon is now is divisible into two swellings, cranial and caudal swellings. So we have to give two more names. The cranial swelling is known as metencephalon and the caudal one is known as, what is the term used for the visualization of a spinal cord, medulla and spinal cord? Myelogram. So that is why this cephalon is known as myelencephalon. So the parts of the brain develop from this cephalon. Telencephalon, two telencephalon are there. So they can give rise to two cerebral hemispheres, right and left cerebral hemispheres. Cerebrum, right cerebral hemisphere, left cerebral hemisphere from the respective telencephalon. Diencephalon is in the midline. It gives rise to all thalami. All thalami means thalamus, hypothalamus, epithalamus, metathalamus, subthalamus, all thalami. The common term used for all thalami is diencephalon. Mesencephalon does not undergo any change throughout. It becomes the midbrain. But the metencephalon gives rise to, metencephalon is the rhombencephalon is the hindbrain. So metencephalon gives, hindbrain means what are the parts? Pons, medulla and cerebellum, these three. Of the three parts, the pons and the cerebellum develop from metencephalon and medulla and its continuation, spinal cord, develop from myelencephalon. So this is the summary of development of brain and spinal cord. First 3 vesicles, later 5. And each one gives rise to these parts. Now you have to label everything. This side they are derivatives. Medulla and its continuation, the spinal cord. So pons and cerebellum develop from metencephalon. Real photograph. Spermatozoon is capable of fertilizing the ovum immediately after ejaculation. Pause. It has to undergo the final maturation process in the female genital tract. What is it called? Capacitation. That is the final maturation process. Capacitation. How much time is required for the capacitation? 5 to 7 hours. So it has to remain in the female genital tract at least for 5 to 7 hours to become fully mature, to become capable of fertilizing a ovum. What about number 2? True or false? True. What about number 3? True. Number 4, no primary oocytes are formed after birth. True, only reduction in the number of oocytes. No extra, reduction only. From 7 million to 2 million, 2 million to 40,000 and 400 finally. Number 5, what is your opinion? This is applicable to 4th degree, correct. Differentiation of the sex of fetus is possible as early as 7th week of pregnancy. You do ultrasonography, can you determine the sex of the baby before 7th week of pregnancy? Then when? Okay, false, then tell me when. When is it possible? See, external genitalia are in undifferentiated state till 9th week of pregnancy. Differentiation begins to occur only from the 9th week and is completed by 12th week. So only after 12th week it is possible to know the sex of the baby. Even after 12th week sometimes difficult because the two thighs are in crossed position, so difficult to see the external genitalia. Only after 14th week it is possible, beyond doubt. So differentiation begins to occur only from 9th week and is completed by 12th week. Only after that it is possible to know the sex of the baby by doing ultrasonography. Identification of sex is possible from peripheral breast smear examination of a normal individual. False or true? False. You see, the polymorph cells, the nuclei of polymorph cells of females present a darkly stained nuclear material called a drumstick body. What is it? This is the inactivated, not bar body, this is the inactivated X chromosome seen in the nucleus of polymorph cells, neutrophils, seen only in females. So you see, when we do peripheral blood smear examination, we can see the drumstick body and we can say whether the sex of the person is female or male by simply noting this drumstick body. Possible. MIF is produced by Sertoli cells of testes. What are the other functions of the Sertoli cells? Support for the developing spermatozoa, supportive function, nutritive function, and blood testis barrier. Blood testis, it acts as the blood testis barrier. So supportive function, nutritive function, blood testis barrier, and production of secretion of inhibin, Mullerian inhibiting substance. Monozygotic twins arise by division of inner cell mass into two primordia. True. See, we have seen the inner cell mass. In the beginning itself, the inner cell mass divides into two inner cell masses. Each one is transformed into an embryo later. That is how identical twins develop. The hard palate is developed from both the frontonasal and maxillary processes. Let us find out. The bilateral cleft lip is due to failure of fusion of right and left maxillary processes. The lens of the eyeball is vascular before birth. The olfactory epithelium is a derivative of surface ectoderm. We shall see the diagrams now. Palate develops from two sources. The anterior part of the palate, as you see here, is known as premaxilla. It is developed from frontonasal process. The rest of the palate, posterior aspect of the palate, develops from two maxillary processes. there are 5 processes in the development of face and some in the development of palate. Look at this picture. Once upon a time our face was like this. This is the frontonasal process. These are the 2 maxillary processes. 2 mandibular processes also are there, but they are fused in this picture. Fused to form one mandibular process. Maxillary and mandibular processes are the parts of first pharyngeal arch. Frontonasal process develops due to development of brain inside. This part of the head enlarges. That is the frontonasal process. In the lower part of the frontonasal process, we see 2 projections. One medial projection, other lateral projection surrounding a small pit, nasal pit. This is the medial nasal process. This is the lateral nasal process. Between the 2 nasal processes, we see the nasal pit, which later becomes the nasal orifice. Now you see, the maxillary process extends from the lateral side. First it fuses with the lateral nasal process. Then it goes further medially to fuse with the medial nasal process. Once it goes further medially to fuse with the medial nasal process, the nasal cavity is totally cut off from the oral cavity and the nasal orifice develops later. Suppose the maxillary process fails to fuse with the medial nasal process. What will happen? Look, the maxillary process has failed to fuse with the medial nasal process. It has fused with the lateral nasal process, okay, but it has failed to fuse with the medial nasal process. What has happened? A cleft lip, and if it goes deep, a cleft palate also can occur. So it results in cleft lip. It may be unilateral or bilateral. In this case, we see a bilateral cleft lip. If it extends internally, it will result in cleft palate also. That we will see. This is the bilateral cleft palate that is extended into the palate also. The premaxilla is totally separated from the other rest of the palate. And this premaxilla protrudes in this part, anterior part of the face. That you can see the photograph now. You can see this is the premaxilla which protrudes. You can see the premaxilla, you can see the bilateral cleft lip extending into the palate. The premaxilla should fuse with the maxillary processes, but the premaxilla has not fused with the maxillary process. It forms a swelling in this region. Bilateral cleft lip and palate. Cleft palate is divided into 2 types: anterior cleft palate, posterior cleft palate. Anterior cleft palate is due to failure of the premaxilla to fuse with the rest of the palate. But posterior cleft palate is something different that we will see. Same thing shown again. You can see the premaxilla which is bulging with 2 clefts here. Same child after some time following surgical correction. Following surgical correction. No trace of it is there. This is unilateral cleft lip. It is seen only on one side. You can see that. Unilateral cleft lip and palate. Now what is the problem here? The maxillary process has failed to fuse even with the lateral nasal process. This has resulted in what? What is this condition? Is this condition? Oblique facial cleft. A cleft extends from the medial angle of the eye. You can see that. from the medial angle of the eye to the upper lip area, to the oral cavity. It is here the nasolacrimal duct is supposed to develop. Due to a large cleft, the nasolacrimal duct fails to develop in oblique facial cleft. The duct extends from the medial angle of the eye to the nose, lateral wall of the nasal cavity. Here such a thing is not possible because of a large cleft, so it is absent. Now this is the posterior cleft palate. Here the two maxillary processes fail to fuse in the midline. There is no bulging in the anterior part. You see that anterior part of the lip and palate is perfectly normal. Teeth have properly developed, but there is a large cleft here, posteriorly. This is posterior cleft palate. This is due to failure of fusion of two maxillary processes. They have fused with the premaxilla, but they have not fused with themselves. Now you are going to see the most severe condition, complete cleft palate, through and through, right from the anterior end to the posterior end. Total cleft palate, both anterior and posterior. The nasal septum you can see here, it has developed properly but not touching anything. Oral cavity and nasal cavity freely communicate. Two cavities freely communicate. Here there is a cleft but not in the lateral aspect of the upper lip. It is exactly in the midline. What is it due to? What is it called first of all? Yes, median hair lip. Failure of extension of tissue from frontonasal processes. The frontonasal process should extend down to form the philtrum of the nose, the red margin of the lip. But it has failed, therefore there is a cleft in the midline. Median hair lip, resembling the lip of the hair. What is it due to? Double nasal cavity. What is it due to? Yes, the right and left medial nasal processes have failed to fuse to form the bridge of the nose. The bridge of the nose is formed by the fusion of two medial nasal processes. They are now separate here. So double nasal cavity. The hard palate is developed from both the frontonasal and maxillary processes. True. Bilateral cleft lip is due to failure of fusion of right and left maxillary processes. Wrong. It is due to failure of the maxillary process to fuse with the medial nasal process. Medial nasal process. Lens of the eyeball is vascular before birth is true. It is supplied by the hyaloid artery during development. After birth, the hyaloid artery disappears, but the canal which transmitted this artery persists in the vitreous humor. That is how we see the hyaloid canal in the vitreous humor. All nerve cells neurons develop from neuroectoderm. An exception to this rule is seen in the development of olfactory neurons. Olfactory neurons, they develop from surface ectoderm. That is why we call them olfactory epithelium. Since olfactory neurons develop from surface ectoderm, they are capable of regeneration. Neurons cannot regenerate. Nerve cell body cannot regenerate. A damage to the nerve cell body is permanent. It's a permanent damage. But olfactory neurons are capable of regeneration because they develop from surface ectoderm, the same ectoderm that gives rise to the epidermis of our skin. So epidermis, continuous replacement of epidermal cells is there. Kidneys start functioning before birth. True. The fetal urine forms the major part of amniotic fluid. We have seen already. The urinary bladder is an abdominal organ in infants. True? Yes, it is an abdominal organ because the pelvis is too small to accommodate the bladder. It becomes a true pelvic organ only around puberty. The pelvic cavity is so small to accommodate the urinary bladder. So it floats in the abdomen. It descends to lie in the pelvic cavity only around puberty. Male urethra gets contribution from all three germ layers. We have seen already one structure which gets contribution from all three germ layers. That is tympanic membrane. The other structure is the male urethra. How, you may ask? The urethral orifice, being a body orifice, should develop from ectoderm. The prostatic part, the trigone of the bladder and the posterior wall of the prostatic urethra, they develop from absorbed part of mesonephric duct. Mesonephric duct means mesoderm. Rest of the urethra is from the endoderm along with the urinary bladder from the urogenital sinus, from the hindgut. So in this way, the male urethra gets contribution from all three germ layers. Tip of the urethra, that is ectoderm because it is a body orifice. Posterior wall of the prostatic urethra, mesoderm. Rest of the urethra, endoderm. The notochord gives rise to the vertebral column. True or false? Yes. Vertebral column develops from sclerotome of somites. Does it contribute anything? Yes. Nucleus pulposus. Even this has been questioned nowadays. There is difference of opinion about this. Some scientists do not agree with this. Notochord giving rise to the nucleus pulposus of the vertebral column. Even that also is not formed from the notochord. That's what they say. So some controversy remains now. The midgut loop undergoes rotation in clockwise direction. Anti-clockwise direction, how much? 270, 3/4 of a circle. The return of midgut loop to the abdominal cavity occurs during 10th week of intrauterine life. First of all, you should know what is returning. Why should it return? From where it should return? Let it be. Congenital inguinal hernia is due to persistence of processus vaginalis. The main pancreatic duct is contributed by both the ventral and dorsal pancreatic buds. Secretion of insulin begins during the 5th month of intrauterine life. True. False. You have to divide the 5 by 2. You will get the answer. 2.5 months. 10th week of intrauterine life. 2.5 months. Now we will see how the midgut develops. The midgut, actually our gut develops outside the body of the embryo, because there is no space inside the abdominal cavity of the embryo to accommodate rapidly elongating coils of intestine. Even that cavity is mainly occupied by the large liver in the right hypochondrium. So liver occupies major part of the abdominal cavity of the embryo, so there is no space for the intestine to develop. Therefore, our intestine herniates into the umbilical cord. It lies outside the body. This is what is called physiological umbilical herniation. It is a normal process seen in all embryos, development of embryos. Once the abdominal cavity becomes sufficiently large, the whole intestine is withdrawn into the abdominal cavity. This occurs during 10th week of intrauterine life. The herniation occurs during the 6th week, and our intestine lies outside the body for 1 month, 4 weeks. Then afterwards it is returned to the abdominal cavity. This is what is called reduction of physiological umbilical herniation. When it returns back into the abdominal cavity, it rotates in the anti-clockwise direction to retain the adult position. -270 in the anti-clockwise direction. Can you read? Oh, it's not there. Colon missing. Development of cecum and ascending colon. See, the cecum lies just below the liver, following return of the midgut loop, as you see in this picture. This is the subhepatic position of the cecum. Then elongation of the colon occurs in this direction. That is how the cecum comes to lie in the right iliac fossa. In the beginning, the cecum is in the 6 o'clock position here. Then the midgut loop cecum rotates in the anti-clockwise direction to reach 3 o'clock position, then 12 o'clock position, and finally to the 3 o'clock, this 9 o'clock position. So it also rotates 270 degree in the anti-clockwise direction along with the midgut. Suppose this last 90-degree rotation does not occur. Let us assume a situation where the last 90-degree rotation does not occur. What will be the position of the cecum? This question has been asked in the last All India entrance examination. The cecum will be found below the stomach in the midline. What is this condition called? That was the question asked. Is it called non-rotation, mal-rotation, mixed rotation, reversed rotation? Yes, it is a form of mal-rotation called mixed rotation. Yes. This was the right answer for that question. Mixed, because rotation has occurred but not complete, so mixed. Cecum found in the midline below the stomach is known as 1, 2, 3, 4. 4 rotations were given. So mixed rotation was the answer for that question. Yes, it is there, mixed rotation. Now the cecum is somewhere here, below the stomach, in the midline. This is the physiological umbilical herniation, as I told you. You can see the bulging, the liver is there, you can see the color, black color. This is the abdominal cavity occupied by the liver. And you can see the midgut here, herniated midgut loop in a 58 days old child, the fetus. After 10th week, when the abdominal cavity is sufficiently large, the whole thing is returned to the abdomen. Suppose it fails to return to the abdomen, what will happen? It fails to return to the abdomen even after 10th week. A child is born with intestines outside. What is that condition called? Child is born with intestine outside. This condition is called omphalocele or exomphalos. This can be surgically corrected. But it should be differentiated from a similar condition. What is it? Gastrocesis. Gastrocesis is due to a defect in the anterior abdominal wall, as a result of which intestines, all abdominal contents herniate due to defect. This is because, as I told you before, the embryonic disc lateral folding, they should come and fuse in the midline. If this fusion is incomplete, a gap may be found through which abdominal contents can herniate. You see the picture? This is the photograph. You see the whole thing is outside now. Gastrocesis. Eventration of the abdominal viscera due to a defect in the anterior abdominal wall, due to failure of fusion of the two edges of the embryonic disc during lateral folding. So midgut loop undergoes rotation in anti-clockwise direction, not in clockwise direction, 270 degree. Return of midgut loop to the abdominal cavity occurs during the 10th week of intrauterine life. True. Congenital inguinal hernia is due to persistence of processus vaginalis. True. Main pylori duct is contributed by both the ventral and dorsal buds. Secretion of insulin begins around 10th week of intrauterine life, 2.5 months. Thus we complete all questions from embryology, but we didn't get time to discuss questions from thorax. That we shall cover in the next class. No problem.